different part of the body.
Is it visible and audible, everybody?
Yes, ma'am.
Okay.
Cancer has distinct molecular or genomic characteristics, often resulting in inadequate response to one-size-fits-all treatment. Tumor profiling is a versatile diagnostic approach that reveals the unique characteristics of a person's cancer, enabling the identification of the most appropriate treatments for that patient and enabling precision medicine. Caris Life Sciences developed comprehensive state-of-the-art molecular analysis tools to characterize the individual patient's cancer and identify treatment options with improved clinical benefit, as well as treatments to avoid. A tumor sample is collected from the patient and sent to Caris, where testing is performed with an overall success rate of 95%. Caris utilizes multiple advanced technologies to assess the fundamental building blocks of the cells, the double-stranded DNA, which stores the genetic instructions, the single-stranded RNA, which decodes the genes and regulates their expression, and the proteins, which are the product of gene expression and have many different functions in the body. This information reveals the tumor's molecular blueprint. Obtaining this large-scale molecular data provides further opportunities to deconvolute the molecular alterations through the application of artificial intelligence and machine learning algorithms. This provides transformative insights into the disease and identifies unique molecular signatures for cancer subtypes, which can predict patient response to certain therapies. Caris is leading this innovation by providing the world's first application of AI in cancer to help guide treatment decisions. The information generated helps physicians identify therapies that would be most beneficial to help cancer patients fight their disease, along with those therapies to potentially avoid. As the leading innovator in molecular science, Caris Life Sciences is actively working to fulfill the promise of precision medicine through unique transformative platforms that help patients, physicians, and researchers improve outcomes and save lives.
Thank you so much.
Thank you, ma'am.
Sorry, everyone. Yeah, thank you so much, Divya ma'am, for sharing such a wonderful animation and video. I think that gave a very good idea about all the testing and tumor analysis, right, guys? Yeah, so ma'am, is your session over? Have you concluded the session, right?
Yeah, yes, ma'am. I'm done with the session, ma'am.
Okay, okay. Thank you so much, Divya ma'am, and it was a wonderful session. What a strong foundation that you have, laid for all our students, and now it will be good for all our other speakers to come in and build more information and more data in that beautiful foundation that you have laid. Thank you so much, Divya ma'am, for that.
Thank you, ma'am.
All right, guys. So with this, we move on to our next session with Miss Kavita Murugesan. So I would like to welcome her. Kavita ma'am, can you hear me?
Hi, Diti ma'am, and hi, Divya ma'am, and hi, the respected attendees who have joined the session. Good morning, everyone.
Good morning, ma'am, and thank you so much for joining us.
Yeah, thank you.
Okay, so before you start the session, I would like to introduce Kavita ma'am to all our attendees. So Kavita Murugesan is working as clinical research trainee with Biotechnica. She's a dedicated clinical research professional with 2 years of hands-on experience in clinical research and clinical operations. With a strong foundation in good clinical practice, that is GCP guidelines and regulatory requirements, Kavita has supported the planning, coordination, and execution of clinical trials across various phases. As a clinical research trainer, she is passionate about sharing knowledge and supporting the professional development of research team with a keen eye for detail and a commitment to quality and compliance. Kavita ma'am aims to foster a learning environment that supports operational excellence and high standards in clinical trials. So she will be guiding you all to the next session where we will be talking about the clinical impacts of cancer genomics, diagnosis, trials, and therapy. So over to you, ma'am. And everybody, as I requested everyone, that please be attentive, note down your question, and so that you can ready to ask your question at the QA session that we will conduct at 3 p.m. Over to you, ma'am.
Thank you, Riti ma'am. That was a nice introduction about me. Thank you so much. And thank you for our previous speaker, Divya ma'am, for such an insightful and an engaging session about cancer genomics. She have explained about all the basics about genomics and tumor development and how we treat cancer genomics, everything. So moving on to our topic. Before moving on to the topic, I would like to share the screen. Just let me know once the screen is visible.
Yes, ma'am, it is visible.
Okay, thank you so much. So, today's topic is the clinical impact of cancer genomics. Good morning, everyone, and thank you for joining the session, by the way. And we are going to talk about one of the most revolutionary changes in the way we understand and treat cancer, that is, cancer genomics. For many years, cancer treatment was guided primarily by where the tumor started, or like where in which part of the body it have developed. So if someone had lung cancer, they were given lung cancer chemotherapy. If someone is having a breast cancer, then they were given breast cancer treatments. But as many of you know, this, as Divya ma'am told also, one size fits all approach didn't always work. Two patients with the same type of cancer treated in the same way could have very different outcomes because each and every individual person's body is different, and mainly the root cause of cancer is completely different. That is why cancer genomics have made up into a revolutionized uh, foundation for the treatment of cancer. Now, some people might not respond very well even after receiving cancer treatment at all. But why does this happen? The answer lies in the genetic blueprint of the cancer, which was taught by the previous taught in the previous session. Today, we know that cancer is not just a disease of the tissue or organ, it's just a fundamental or a disease of our DNA. Mutations, rearrangements, or let's say alterations in our genes drive cancer growth and determine how it behaves. So, let me take you to a session where we can able to explore what exactly cancer genomics is and why it matters. So, how it impacts diagnosis, prognosis, treatment, and even prevention of cancer growth in the human body, and how clinically we are going to deal with the cancer genomic therapies, like how clinical trials are being redesigned around genomics. So, there are certain set of regulations so far in the clinical trials and in the clinical research field, but when it comes to cancer genomics, how the clinical trials are being redesigned for genomics with special trial types like basket trials, umbrella trials, and etc. And whenever we talk about clinical trials, we need to be look into the ethical and regulatory considerations because we are dealing with a person's genetic information, which is very sensitive, and also we should be the primary responsibility should always consider the patient's safety and well-being. And at the last, we will look into some real world applications, how hospitals, pharma companies, and even AI are shaping the future of cancer care. Maybe by the end of this session, I hope you will not only understand the science but also appreciate how genomics is changing lives every single day. Let's begin by the foundation. What exactly is the cancer genomics? So, what is exactly a clinical cancer genomics? Clinical cancer genomics is essentially about applying the genomic knowledge directly to patient care. Now, what does this mean? It means that the genetic information we uncover from a patient's tumor or even their inherited DNA can actually guide how we diagnose cancer, how can we classify it more accurately, and most importantly, how we choose the right treatment here. So, in practice, clinical cancer genomics involves in the analyzing the DNA and RNA of each and in every individual cancer patients, like involving the analyzing of DNA and RNA of the patient's cancer cells. By doing this, we can detect specific genetic alterations. These alterations give us several valuable insights. First, they can help us predict how well a patient respond to a particular therapy. For example, let's take if a mutation indicates sensitivity to a targeted drug, that drug could be a better choice than standard chemotherapy procedure, right? And secondly, genomic analysis can reveal inherited cancer risk. This is important not only for the patient but also for their family members who might share that genetic predisposition, right? And thirdly, clinical cancer genomics about moving from generic cancer care to truly personalized care. Like as Divya ma'am told, the right patient with the right drug at the right time based on the tumor DNA. So precision medicine means right patient, right drug at right time. But here in cancer genomics, based on tumor DNA, we are going to give the right patient with the right drug at the right time. So now, uh, why is this important? Let's break it into four key areas, like the importance of cancer genomics in clinical trials. What we exactly do in this. The first and foremost step will be diagnosis. By analyzing the tumor's DNA, we can identify specific mutations that help us classify the cancer more precisely. For example, let's take two people, may be both diagnosed with lung cancer, but if one has EGFR mutation and another has KRAS mutation, maybe different types of genetic mutations, these are actually two very different diseases genetically. We can't give the same kind of treatment to two different patients having different types of gene mutations, right? So in cancer genomics, the first and foremost step and the first purpose would be the diagnosis process. And the second process, based on proper diagnosis, we will be moving on to prognosis, like predicting disease course. Like some mutations tell us about how aggressive, how intense the cancer might be. Like for instance, the TP53 mutation linked to uh, cancer is often associated with poor outcomes because it removes one of the body's key protective mechanisms against cancer. So we should be predicting the intensity of the cancer before moving on to the treatment that is going to be given for a specific patient. Thirdly, the treatment procedure. Like this is where exactly the precision medicine comes. Like precision medicine really shines by knowing the mutation. We can select drugs that specifically target it. Let's take example of HER2 positive breast cancer here. Like, have you seen so many women with HER2 positive breast cancer can be treated with a specific drug like trastuzumab. A targeted drug exactly works only in on those patients. But imagine if if a patient is getting a breast cancer without having this kind of HER2 positive breast cancer or HER2 positive protein inside that, how does exactly this treatment is going to work on that specific person. So, without knowing the genomic testing, without knowing the baseline causative agent, or without knowing the root of the cancer treatment, we couldn't able to proceed with the treatment procedure. So, whenever we are giving treating the cancer patients with the specific precision medicine, we should be selecting therapies targeting specific mutation, which kind of mutation happen to which set of population, like that. uh, and at the last, prevention, like genomic doesn't just help once cancer has developed, it can also identify inherited mutations that increase a patient's risk. Maybe yeah, let's take a famous example, is the BRCA1 and BRCA2 mutations, which significantly increase the risk of breast cancer and ovarian cancers. Many of you may recall a famous actress like Angelina Jolie publicly shared her decision to undergo preventive surgery after testing BRCA1 mutation. That was really a direct example of cancer genomics saving lives through prevention, like detecting the mutations in the preventive stage, and we can go to the preventive surgery before even the cancer is occurring. So I would like to say, like, cancer genomics is basically the understanding of cancer by decoding its genetic blueprint. By decoding this, we can give the patient with a standard care and a precision medicine and a personalized medicine for each and every patient. So, in summary, I would like to conclude that a cancer genomics is about the reading the cancer's DNA and using information for diagnosis, prognosis, treatment, and prevention of each and every individual, that we call as precision oncology. The right patient, right drug at the right time. Now that we know what cancer genomics is, let's talk about this how or why this approach matters so much in today's world, why it actually matters. And well, because it gives us something we never had before, the ability to treat cancer smarter, not harder. Like, I would like to give you a few examples here. Like, in the past, treatments like chemotherapy were often compared to a carpet bombarding strategy. They kill rapidly dividing cells, but unfortunately that includes healthy cells too, right? That's why patients experience side effects like hair loss, nausea, vomiting, very low immune response after taking any cancer treatments, or after taking any cancer drugs or any cancer medicines, right? So let me give you examples of cancer genomics in actions, like taking lung cancer for EGFR mutations treated with EGFR inhibitors. So these kind of inhibitors will directly stop EGFR mutations and help to prevent or reduce the intensity of cancer. And ALK rearrangements treated with ALK inhibitors. So this will help to rearrange or arrange the ALK set of genes in a proper way, so that the occurrence of lung cancer will not be there. Then for breast cancer, HER2 amplification treated with HER2 targeted drugs. So these set of targeted drugs will help the breast cancer to get reduced, or the intensity of the breast cancer might reduce. Likewise goes with the melanoma, like BRAF V600E mutation treated with BRAF inhibitors, and colorectal cancers like KRAS mutations with specific set of anti-EGFR drugs. So these set of inhibitors and these set of targeted drugs will help directly go and target on the specific set of mutations and amplifications, which helps the patients to give a personalized and a precision medicine. But over the last decade, technology has completely transformed cancer research and care. Let's move on to the technologies that we use in the cancer genomics over the last decade. Like, the technology completely transformed, as I told you. uh, we can sequence a cancer's DNA in ways that were unimaginable 20 years ago. The most commonly used tools is next-gen sequencing, whole genome or exome sequencing, and RNA sequencing. The next-gen sequencing, like think of NGS as a super fast and super accurate way of reading the DNA. With NGS, we can detect mutations, gene amplifications, and even genetic fusions. For instance, if a lung cancer patient has an ALK fusion, NGS can detect it and the patient can be given ALK inhibitors. Like, crizotinib is one of the ALK inhibitors we used to give for lung cancer patients. Let's go for whole genome sequencing. In whole genome or whole exome sequencing, WGS basically it looks like the entire genome code. Imagine reading all 3 billion letters of DNA. WES focuses only on the coding regions, the exons, which make up about 1 to 2% of our DNA but contains most of the disease causing mutations. Another powerful tool is RNA sequencing. This doesn't look at the DNA itself, but at how genes are being expressed. It basically helps us to understand which genes are switched off, which genes are switched off in the cancer, which particular gene is causing the cancer, which expression patterns predict how a tumor will respond to the treatment. So all these set of technologies are used in clinical trials for treating cancer genomics and for detecting and for analyzing or for making any decisions before giving any cancer treatment for a patient. Next, we will be moving on to genetic testing. So, role of genetic testing in cancer risk prediction and personalized medicine. So, genetic testing. How do you actually test a gene has certain problems or a tumor is caused because of these kind of mutations or not? So there are two types of genetic testing. One is germline testing, identifies mutation passed from parents that increases lifetime cancer risk. And the second one is somatic testing, identifies mutations only in cancer cells, guided it helps to guide treatment. Like this germline testing is helpful for identifying all set of mutations passed from our parents, whereas this somatic testing helps to identify mutations in cancer cells, like which particular area or which particular tumor the cancer has been occurring or not. Based on this risk prediction and the genetic testing, we will be applying these kind of results from this germline testing and somatic testing. We will be moving on to the risk prediction, personalized therapy, and pharmacogenomics. Like, what is risk prediction? Risk prediction is basically helps to identify, like this means looking at the inherited or a germline mutation that is born with, example, people with Lynch syndrome carry mutations that put them at much higher risk for colorectal cancers and certain other cancers also. If we identify these individuals in the earlier stage itself, we can start regular screening, lifestyle changes, or even preventive surgeries. This literally saves lives before even the cancer appears, particularly in children, those who are going. These days, cancer is occurring at very young age also. So, using this kind of risk prediction technologies, we can help the children to get not only the children, even the adults to get prevented from the cancer. Secondly, a well-known personalized therapy, like matching the treatments to the genetic alterations. Like this decision is based on the genomics, the genetic like genetic material that is causing any alterations, like the right time and the right drug for the right diagnostic process. So, personalized therapy has been like paving a way to cancer genomics for treating the patients with certain kind of mutations. Then pharmacogenomics, like adjusting the doses based on metabolism genes. So these are certain testing procedures that we basically use in clinical cancer trials. Now moving on to the cancer trials, like here I have given some examples like BRCA1 mutations, higher breast or ovarian cancer risk, PARP inhibitors improves outcomes, and Lynch syndrome. Here I have taken colorectal endometrial cancer risk managed with frequent colonoscopies, and KRS wild type colorectal cancers eligible for anti-EGFR therapy. So here are certain examples that I have mentioned. Then just give me a minute, guys. Like genomic driven clinical trials. So far we have seen about what is a cancer genomics, how we are going to treat, how we are going to detect the mutations happening in a cancer cell, or personalized medicine for individual and for a set of audience exposed to same set of mutations. So there are so many things we have seen right about cancer genomics. Moving on to the main topic about of today, like genomics driven cancer clinical trials. So the evolution of clinical trials are like is enormous, and the traditional clinical trials we used to enroll the patients for cancers based on the cancer type. Like we will be taking a set of breast cancer patients, we will be taking a set of lung cancer patients, we will be taking a set of blood cancer or a melanoma patients. But imagine now this cancer genomics is driving the clinical trials into the next stage. Like we will be taking patients or enrolling the patients based on mutational signature, not the tumor location. That is the causative agent for the cancer. We used to enroll the patients based on the causative agent and not on the tumor location. Let's look into the designs, what kind of genomics or how the genomics are involved in the clinical trials, and what are the trial design types, what are these trials are particularly for. Let's look into basket trials. Like this based on the cancer genomics, this FDA and the regulatory authority bodies have designed certain types of clinical trials. One is basket trials, one is umbrella trials, and the other one is adaptive trials. Apart from this, there are separate other clinical trials which we usually perform traditionally. But these three are evolving in today's generation and today's modern clinical trials, and paving the way for healthcare to bring advancements in cancer therapies and treatments and in the diagnostic procedures. So let's look at each and every trial design type first. The first one is basket trial. So as you can see, basket trial is a type of clinical trial designed where patients are grouped, put in the same basket. So just imagine yourself, like you are going to be a sponsor and you are going to be selecting a patients for certain clinical trials, and you have a specific hospital site for conducting a clinical trial. Now, instead of doing a routine clinical traditional trial for cancer, the sponsor have taken a set of basket. A sponsor has taken a basket in which the common causative agent, that is genetic mutation, the common causative agent for most of the cancers is genetic mutation, right? So now the patients are put into the basket, that is put into the basket based on a common genetic mutation or any molecular marker rather than the organ or tissue where their cancer started. Traditional cancer trials are basically organized by tumor site. As I told you, based on lung, breast, colon cancers. But what we do in this basket trials means which it will be organized by molecular alteration. So let's take this BRAF genetic mutation. So in the basket trial, instead of using the tumor site, we used to take this BRAF mutation patients. We used to segregate these set of patients, whether they can have a melanoma, or whether they can have a colon cancer, whether they can have a thyroid cancer. But we used to take this set of particular genetic mutation patients, and we used to give a targeted therapy, that is a specific kind of treatment, BRAF, VAE, regardless of tumor location. We used to give a proper inhibitor to treat this BRAF in alterations or molecular alterations inside the patient's genetic information or a genetic formation. So this BRAF inhibitor will go and act into each and every patients who are exposed to these particular set of genetic mutation, and it will act as a therapeutic agent for these kind of patients. So instead wasting so much amount of instead wasting so much amount of money and time spending patients for melanoma separately, colon cancer separately, and thyroid cancer separately, if we can able to find out the genetic mutation, or if we can able to find out the uh, if we can able to find out the root cause of any particular cancer, regardless of the site that they are appearing, we can easily able to give a targeted therapy, and we can able to see a positive outcomes and the positive statistical results, and the recovery survival rate, overall survival rate of the patients. We can able to see so many improvements and increased survival rates in the patients having this kind of genetic mutations. So in simple words, I can explain you, in basket trials, we will be finding out the causative agent for a particular trials or a particular set of genetic mutation, and we will be putting all those patients in the basket, and we will be giving a targeted therapy. This can the main advantage, or this can help the patient to get the right therapy, and it can save the time of the patient, it can save the time of the sponsor time and the cost of the sponsor also. It can lead to bring more revolutions in modern clinical trials. Secondly, we used to have this umbrella trial. So what is this umbrella trial basically? As the word suggests, you can easily able to find out based on the cancer type. Like an umbrella trial is a type of clinical trial design where patients with the same type of cancer or same tissue or organ origin are divided into subgroups based on their molecule or genetic alterations. So let's take genetic mutations on one side. Now patients with genetic mutations are having certain kind of issues, but those patients having a different type of cancers like breast cancer, lung cancer, colon cancer, but the genetic mutation, the causative agent or the culprit is one person. So now we are going to inhibit that culprit, and we are going to treat that. That is considered as basket trial. Moving on to the umbrella trial. What is exactly a umbrella type? As you can see in the image or the pictorial representation, one umbrella with so many markers like EGFR mutations, ALK rearrangement, KRAS mutation, and PDL1 overexpression. So these are certain genetic mutations or any kind of molecular alterations happening in a same set of organ. Like lung cancer may have more. See, let's take a four different type of person for who is like those who are exposed to lung cancer. Okay. Do you really think that all these lung cancer patients will have the same set of causative agents for getting those lung cancer? See, one set of patient may be a chronic smoker, one may be a teetotaler, one may be exposed to smoke or work in any kind of chemical industries. So the exposures, the exposure, the causative agents may be different, right? But in umbrella trials, what we used to do is we will be taking, let's take, the sponsor is going to study about the lung cancer. Means what the sponsor does here, they will be allocating, or they will be grouping the patients in different types. This lung cancer patient, what is the causative agent? They used to find out the molecular and genetic alteration, so that this particular, let's take EGFR mutation is happening. What this EGFR mutation patient should receive, they should receive this particular inhibitors. We cannot treat them with ALK inhibitors or KRAS inhibitors, right? So based on the checkpoints or the different types of causative agents, where a particular is exposed to one organ cancer, that is lung cancer or a breast cancer or anything, any type of cancer, right? So basically umbrella type is taking one cancer with different causative agents and treating all the uh, causative agents by giving different types of targeted therapy. In basket clinical trials, many cancer the same reason, but in umbrella cancer trials, we can able to see one cancer with many reasons. Based on this causative agents, we will be treating them with different set of medicines. In basket trials, we used to give only one targeted therapy, but in umbrella trial, we used to give so many set of targeted therapies based on the causative agents where the patient is exposed to the cancer. Basically, the umbrella is one cancer type with different treatment arms under. So, uh, yeah, let's move on to adaptive trials. This is one interesting topic, not only for cancer genomics, but for the rest of the clinical trials. All the clinical trials involved in current modern days clinical trials are undergoing into adaptive clinical trials. As the name suggests, what is actually adaptive trial? Like these trials are designed to learn as they go. Like adaptive trials mean smart trials that learn as they go. So what is exactly this adaptive clinical trial? In traditional trial, the protocol is fixed. The sponsor used to prepare a protocol, the drug, the number of patients, the endpoints. Nothing will be changed, right? Nothing changes until the trial ends. But what if a drug is clearly not working, or what if one arm is showing great results early? Shouldn't we able to adapt to the better side, or should we continue with the same protocol that we prepared at the beginning? So difference between the normal trial versus the adaptive trial, a sponsor used to prepare a set of treatment before, even before the trial starts, and the sponsor used to give the treatment for a certain number of years, and at a certain different phases. And at the end, the sponsor used to collect all the results, and after that, the sponsor will be submitting to the FDA process, collecting all the results, and based on the results on based on the outcomes, either the drug will get approved, or the patient might shown any improvement, or if the drug may not be approved if the patient faces any drastic serious adverse events or adverse events, right? This is the standard clinical trial procedure. But what we do in adaptive clinical trial, so this adaptive clinical trial is not only for cancer genomics, it is paving way for all the other cancer related, all the other therapeutic areas, not only particularly in oncology field, but particularly in oncology. What we actually do in adaptive clinical trials, they will basically allow modifications can be done during the trial based on the interim results. What is interim? Any changes occurring in the middle of anything. So based on interim results, like without compromising the scientific integrity, the ineffective arms, that is ineffective or a no improvement patients, will be given treatment like another alternative options. If treatment A is not working, then we don't have to waste time, or we don't have to risk on patient life. We should be formulating with treatment B. We should be treating them with treatment B. If treatment B is also not working, we can adapt or we can modify. If some set of patients are facing certain adverse events using treatment A, if certain patients are using or getting benefits using treatment B, then what are we supposed to do here? We can give the same treatment that we are given for treatment B to even treatment A patients. So adapting or adjusting or giving flexibility in the trial during the clinical trial or during the or in the beginning of a clinical trial can be encouraged. And interim analysis guides changes, maintain trial integrity with pre-specified rules, and reduce time, cost, and patient exposure to ineffective treatments. So yes, right, if something is not working, what we usually do, we will be going to some other plan. If plan A is not working, we used to go to plan B. If plan B is not working, we used to go to plan C. Like that, the sponsor in adaptive trial, the sponsor will try so many treatments and will adjust or will modify the treatment procedures based on the improvement shown by the cancer patients. So this is adaptive trials in oncology field. So moving on to next important topic. Uh, just give me 1 minute. So when we talk about clinical trials, what is the first and foremost thing that we used to be considering? Ethics and regulatory approvals. So before starting any clinical trial, the ethical and regulatory considerations should be there in clinical trials. Without ethics and regulatory approvals, we cannot able to proceed with clinical trials because we are going to deal with patient data, patient safety, and patient well-being. Without regulatory and ethical considerations, we cannot able to proceed or we cannot able to risk patient lives, right? So maybe this will help to this raises ethical and regulatory challenges when we are performing any adaptive trial design or any basket trial or any umbrella trial. But what do we do in this exactly during this ethical and regulatory considerations? We used to be, while genomics advances cancer care, it raises unique and ethical regulatory issues like incidental findings, discovering unrelated mutations like BRCA in lung cancer tests. Should this be disclosed to anyone? Can we disclose this patient data to anyone? Or privacy, like genomic data is highly identifiable because a person's genetic information is extremely sensitive and nobody can able to disclose to any person, right? But getting involved in clinical trial, it is very challenging without disclosing the patient data to some other person, because so many people will be involved in clinical trial and we need to maintain the ethical standards in clinical trial, right? So this will mainly help to save the information of patient and protect the or maintain the confidentiality of the data of cancer patients. Then equity, high cost of testing, like limited access in low resource settings. See, let's take basket trials as an example. We are going to hire so many number of different set of cancer patients, right? Like lung cancer, colon cancer, breast cancer. So though the genetic mutation or the causative agent may be the same, we need so many set of different patients from different regions. Imagine the uh, cost or the amount of cost involved in this particular trial. No, we need different set of cancer centers from different states or different regions. Then we need different set of treatment procedures, or we need different set of diagnostic procedures for each and every patients. So limited access in low resource settings. So this eventually this will be a challenging one for getting regulatory approvals. Then informed consent. Patient must understand implications of how whole genome testing. In India, not only in India, if we go and if we tell patients, that is not cancer patients, if we go and tell to normal patients that we are going to uh, like if we are going to treat you for certain trials or clinical trials, we are going to test some new drug on you, will you really think the patient will directly accept and actively involve in clinical trial? No way at all, because nobody will actually believe in standard care like alternative therapies than standard care treatment. If someone is taking diabetes medication for so longer time, that patient will not be cooperative for getting into another set of treatment as an alternative therapy. We will be giving a new set of treatment for you in this particular clinical trial. So those patients will not agree to that, and they won't be signing the informed consent. Basically, what is informed consent? Getting consent from the patient for giving any new treatment to the patients for in a clinical trial. The patient should be informed of all the things that we are going to do in this clinical trial, like that. So this applies to normal patient, but imagine a cancer patient. No, a cancer patient will not see. First of all, exposing to cancer itself will be a very challenging thing for the patients. Now going to them and explaining how this whole genome testing will happen, how this treatment procedure will go on, how this adaptive trial, if A is not working, we are going to give you B, like these kind of factors when we mention to the patient, will you think the patients will agree to that? This is again a very challenging thing, but the patient should be given consent before starting any clinical trial. These are certain regulatory consideration which were put by the regulatory bodies, that these kind of or these set of uh, informed consents should be given to patient under implications of whole genome testing. So these are certain regulatory considerations which needs to be resolved before even starting the clinical trial. Again, it's some ethical and regulatory challenges for the sponsors to bring new innovations in clinical genomics. How they can actually prevent, or how they can actually stop, or how can they face this kind of challenges? They have to be well prepared with advanced set of data. They have to be submitting a set of preclinical data and a solid evidence for a precision medicine or a personalized medicine, where though the drug is going to be tested new for the patients, but they should be having a prehistoric data about or any literature survey or any literature data would help them to get the approvals, and consideration can be easier for them. So these are certain things. Now moving on to regulatory landscape. Even after having this many challenges, how this regulatory landscape actually works. Firstly, whenever we approve a targeted drug, whenever we are moving into or we are going to get approval for any targeted drug, the FDA requires approval of companion diagnostics like HCR to IHC test for this kind of drug. So this kind of companion diagnostics is very important for getting any regulatory approval when doing a clinical trial in USA. This needs to be get before even starting the trial. We should not be waiting until the end of the trial. Even when we start the trial, we need to be getting a proper regulatory approval from the FDA when you are going to perform any clinical trial in USA. Let's say in EMA or any European region or ICMR for India, guidelines on genetic testing. So there are set of genetic testing guidelines. The patient should be tested in this kind of way. The patient should be given informed consent in this set of way. The patient should be having this set of data with them. The patient should not be exceeding this age limit. Only these kind of patients should be taken for genetic testing. No vulnerable population should be taken for genetic testing. So there are so many genetic testing guidelines drafted by EMA, ICMR and FDA. So all these guidelines should be followed before starting the clinical trial by the sponsor, and proper consent. As I told you, a proper consent form should be there, and a proper consent should be given to the patient, because we are going to use their genetic information, right? We should not be misusing or anything, any we should be protecting the data, and we should be maintaining so much confidentiality about patients data in genetic testing. So the sponsor should follow all these guidelines on genetic testing when doing a clinical trial in cancer genomics. Then GDPR treats genomic data as, so this is a data protection right for European region. So treating any patients with genomic data as sensitive personal data. So the sponsor has to be follow this GDPR before starting the trial, and the sponsor has to follow these guidelines drafted by GDPR when treating genomic data with very sensitive manner. Like examples, 23andMe controversy. Direct to consumer genetic testing raised concerns over accuracy and interpretation. So this set of controversy, no, this controversy have raised to, like, they have literally questioned the sponsor, like how can you directly go and do genetic testing on patients without their consent, like without their opinion or without their value, without, some patients may not have knowledge on genetic testing, but this act will help the patients, or will help the trial participants to protect their data and raise concern if they face any kind of leaking of any personal data, or if any confidentiality matters, they can raise concerns to the government of each and every country. And then Myriad Genetics case. This is one case where US Supreme Court, no, like in 2013, ruled that naturally occurring genes cannot be patented. So these set of genetic inhibitors and these set of genetic information cannot be patented, and it should not be used under any, like any serious sponsor companies without having a proper consent, or without giving a proper information to the patients. So this is how a, a regulatory landscape, and this is how regulatory guidelines are drafted for cancer genomics in clinical trials when treating genetic information of any patients. Then moving on to the real time applications. So what is exactly real world applications? Now let's step into the real world. How is all this being applied in hospitals and the pharma industry today? What we are doing with all these clinical trial designs, all these clinical trials in today's world in hospitals? We now have something called molecular tumor boards. These are multidisciplinary teams, like multidisciplinary oncologists, pathologists, geneticists, interpret, bioinformaticians. So there are so many, right, who sit together, review a patient's genomic profile, and decide the best treatment strategy. So you can have how this clinical trial is going to directly implement in real world applications. So this is how it happens. Molecular tumor boards will be organized in separate cancer institutes or cancer related hospitals. They will be organizing a set of multidisciplinary teams. They will be consisting of oncologists, pathologists, geneticists. These kind of teams will be interpreting this genomic data obtained from the patients for treatment procedures, and they will be taking which kind of treatment should be given to which set of patients. So let's take an example of Memorial Sloan Kettering Cancer Center developed by MSK Impact. So this is one example I have taken here, a test that analyzes 468 cancer related genes. Patients tested with this are often matched to specific clinical trials or therapies based on their mutations. So this this is how each and every hospitals or each and every foundation, like foundation 1 or garden 360, so there are so many panels like this. They used to have this set of multidisciplinary teams, and they used to take actions like how the treatment procedure should be going on, how should we move on with the tumor procedures, how should we detect this patient, how should we group this patient, how should we uh, organize or collect these patients. So like that it used to go on. So moving on to pharma industry, companions diagnostics like developed alongside drugs. So in pharma, genomic testing has become central to drug development. Companies no longer just make drugs for lung or breast or any specific cancers. They make drugs for the mutated genes. They make drug for specific targeted therapies. They make drug for rearranged lung cancers or any rearranged uh, implications in genetic testing. Then companion diagnostics is one of that and are developed alongside drugs. Then biomarker based on R&D pipelines now focus on druggable mutations. Like the R&D is now getting into research and development of druggable mutations. Which kind of mutations is happening in which particular gene, and we should be providing drugs for that particular mutations. Then liquid biopsies, exciting development. As a previous speaker told us, blood based detection of circulating tumor DNA, that is ctDNA, for monitoring responses. Instead of invasive tissue biopsies, a simple blood test can detect tumor DNA fragments circulating in the bloodstream. For example, the company Guardant developed like so I have told you, right, the company Guardant have developed a test to detect the EGFRT7090M mutation in lung cancer. If this mutation is found, patients can be treated with drug like this osimertinib therapy. So if this for this mutation they have developed this therapy. So regardless of which organ it is going to affect, this kind of mutation is given this therapy. It can cause lung cancer, it can cause breast cancer, it can cause colon cancer. Whatever the therapy is, it can be treated with it can be treated with this kind of therapy. So this is how these are very few examples that I have taken from hospitals and pharma industries. So you can refer to internet for there are so many other examples, and there are so many other clinical trials who have shown promising results in cancer genomics. Then moving on to growing demand in clinical research and industry perspective. Maybe in clinical research, like you can look into like surge in precision oncology trials, like the developing or the growing need for the basket umbrella trials and the adaptive trials, growing need for bioinformaticians and data scientists and trial designers. Just imagine, no, if you could able to read, if you could able to know these kind of information, these kind of growing needs can be met. You can become a bioinformatician, you can become a cancer data scientist, you can become a cancer trial designers. So there are so many growing demand in clinical research industries for this particular clinical trials. When you have this certain idea, or when you have this set of data with you, when you have certain knowledge in this, you can get into this kind of thing. Or else, if you are from a background of pharma or any chemical or any life science background, you can enter into industries like pharma industries using genomics to identify targets, how to identify how to support drug development, how to identify new targets like these kind of inhibitors, or how to develop tumor agonistic drugs. So you can refer to as many. This is not only for cancer industry, it applies to all industry, but the growing demand in cancer industry is huge. You can literally enter into pharma industry by knowing the drug development process, like identifying the lead, identifying the compound, like drug development, drug discovery. So by knowing all these factors, you can easily enter into these set of fields and future trends like AI in genomics, AI plus genomics, AI tools interpreting sequencing data. Companies like IBM Watson, Google, Deep Variant, these companies are basically supporting AI plus genomics. You can enter into these kind of you can enter into these kind of companies for future developments. So these are going to be the future, guys. I like no, nobody is no one is ready to waste their time or waste their amount cost by uh, going into traditional trials. These trials are evolving. As this evolution occurs, people are looking for emerging trends and looking for promising results. And this AI is paving way for genomics and cancer related trials to give a better outcome and to give a better output for all kind of peoples. Secondly, population genomics. UK around 1 lakh genomics projects, particularly for genome testing alone. UK has been spending so much of amount, and India Genome Valley. So these kind of genomics related uh, works or genomic related revolution, you can easily enter into and you can get easy jobs in this field. And this is really a great revolution, right? Because see, working under genome project or working under any particular these initiatives basically aim to build a database of genetic information that can be used to predict risk, develop new drugs, and personalized care for entire population. So these are literally paving way for the next generation to enter into cancer genomics and cancer related clinical trials. Then real time monitoring, as we saw in the last lecture, like liquid biopsy is evolving rapidly. Soon we may able to monitor patients in real time for cancer relapse just through a blood test, even before the tumor shows upon. See, from an industry perspective, sorry guys. Oh, sorry. From an industry perspective, if we take no, companies are now focusing heavily on genomics. Drugs are being developed for previously undruggable targets like many KRAS inhibitors, and there are so many inhibitors. So this means a new career opportunities for you all guys. Around 200 plus students are here. There are new career opportunities for you all guys opening up in bioinformatics, clinical research, trial design, and data science in oncology fields. The demand for people who understand both genomics and clinical research is growing faster than ever. FDA is approving over 40 oncology drugs in clinical trials approved with genomic biomarkers from 2017 to 2023, and Novartis, Roche. So these are certain leading pharma companies. So companies like Novartis, Roche are playing integrating genomics into oncology pipelines, and hospital practice like Tata Memorial in India and Mayo Clinic in USA integrating genomic tumor boards. Like, see, these are your future opportunities, and this is how the cancer genomics have been developing and paving way for you all to get into this field. You can get into this field from the start by knowing the basics of cancer genomics itself. Like, I would like to conclude. So as we come to the end of this session, let's recap. Uh, I would like to give you a few pointers. Cancer genomics has transformed our way of understanding cancer. We no longer see cancer as a disease of the organ. We see it as a disease of the genome, right? It impacts every step of cancer care, like diagnosis by identifying the mutations, prognosis by predicting the outcomes, treatment by through precision medicine, and prevention by detecting inherited genes or inherited risks. Then clinical trials are evolving with designs like basket trials, umbrella trials, and adaptive trials, ensuring that new treatments are tested smarter and faster. We have now seen how hospitals, pharma industries are already applying these principles in real life. And we have looked at the future, right? Where AI, population genomics, and liquid biopsies will make cancer care even more personalized. The key message I want to leave you with is the future of cancer treatment is not just about fighting cancer. Believe me, guys, it's about understanding its genetic fingerprint and targeting it with precision. Uh, Thank you so much for your attention. I hope this session has given you not just knowledge but also inspiration about possibilities ahead in the future about the cancer genomics. And yeah, uh, there is a separate Q&A session, and I would like to answer a few questions that have been put forward now. Yeah, you guys will be having, and not at the same time. All these markers will be applied at different patient at specific timings. It will be designed in the protocol. So generally, whole genome sequencing can take anywhere from a few hours to several weeks, but this process will help us to speed of sample processing, DNA sequencing technology, data analysis speed, and like purpose of sequencing will be very fast. So that's why we use whole genome sequencing. And can the circulating DNA? Yeah, it applies to all set of cancers in the modern healthcare revolutions. Thank you so much, Nagendra. Multiple targeted therapies under one but if the same patient has. So basically, a patient is going to have a certain cancer for one particular genetic mutations. There are very few cases where multiple mutations occur for patients for certain treatment, but there are certain adaptive trial designs are now working by the sponsor companies or the pharma companies where the multiple mutations can be treated. We will be prioritizing which therapy arm they should enter based on the intensity of the mutation, which mutation is rapidly growing, based on which mutation the patient has exposed to this kind of cancer, which mutation is very intensive. Based on this, we will be working on taking the patient into treatment arm. Then, ma'am, can you talk about oral cancer? By so these kind of lectures will be taken by some other faculty. But oral cancer, we used to find out the causative agent and we used to find out the mutations and genetic causes, and then we will be treated them with particular inhibitors. Yeah, so the entire genome sequences will be taken for detection process and like diagnostic procedures. So let me give you an example. If a person's genetic information, how someone can misuse genomic data? So this is a very quite interesting question. So I will give you an example. Basically, take one person, no? Take one person like take someone is going to involve in genetic testing. Okay, it basically creates tension within a family because the results can reveal information about other family members in addition to the person who's tested. Then the possibility of genetic discrimination in employment is also a concern. Thirdly, if a person's genetic information is known by some other person, they can do anything with that person. They can misuse the data by giving it to some other person. If a person's gene have any kind of specific characteristics, then they can be used the data with some other persons also. So these are certain things that leads to misusing the genomic data. So can repetitive DNA sequence can. So, Shruti, Subhash, I didn't get your question exactly. Can you please elaborate or can you please explain me your question again?
Kavita ma'am, I'm sorry to interrupt. So are you done with the session, ma'am?
Yes, ma'am, I'm done with the session.
Okay, okay. So actually, I see so many questions and queries are there. So I just wanted to tell all the students that, as I mentioned, that we will be having separate Q&A session at the end also. So guys, please do not worry that your questions are not getting answered. Ma'am will be joining for the QA session, okay? Because like it's 12:30 and we have entered our scheduled lunch time. Kavita ma'am will be answering all your questions in the QA session. Okay, so just hold on to your question, and at the QA session, you can ask our faculties. Thank you
Thank
so
you.
much,
Thank you
Kavita
so
ma'am.
much, Diti ma'am, and thank you so much everyone for attending this informative webinar or workshop. So thank you guys. Thank you so much, Diti ma'am.
Thank you, ma'am, and all thanks to you. You did such a good interactive session. I can see so many questions people asked, and you answered all of them patiently. And I hope all of our attendees got a good idea about how clinical research is nowadays, totally data driven and helped by the driven by the information that we get from NGAs and various genomic analysis, and how actually this clinical trials are getting conducted nowadays. Thank you, Kavita ma'am, once again. And I'm looking forward to see you again at the QA time. All right, guys. So being said that, we will take a short 30 minutes lunch break. So I will stop the session here. I request everybody to go ahead, recharge yourself. Okay. So take a good but nutritious but healthy lunch, okay, so that you don't feel sleepy for the next sessions, because next sessions are again power packed. So the session right after lunch will be taken by Dr. Nilofer, and she will give you an idea about how artificial intelligence is helping us in this area of cancer genomic studies and research, and followed by the very important thing, or thing that for which many of our attendees have joined us today, to get a clear perspective on various career pathways, or the projections, or the different career paths that a person can take in this particular field. So with that, Somrita ma'am is ready for the session. Okay, so we'll take a 30 minutes break and see you all at 1 o'clock. Thank you, everyone. See you all at the post lunch session. Hello, everyone, and good afternoon once again. No, Shikha, we have not ended. We were just taking a short 30 minutes break for lunch. And here we are again. All right. So are you all ready? Did you all get your lunch? Lunch done, everyone? Okay, great. All right. So I'm just saying that number of attendees have actually reduced a little bit. I think people are yet to join from lunch. All right. Okay, so anyways, we are on a tough schedule, as I have mentioned, all of you, and we cannot wait a lot. So let the people join in between. And we are ready to start with our session after lunch. Okay, so in our post-lunch session, first session, that is ideally the third session of the day. So there we are looking at the application and also preparation for artificial intelligence in corporate all right. So for that, I have Dr. Nilofer with me. Nilofer, thank you so much for taking time on this Saturday afternoon.
Ma'am, your voice is not clear.
Am I audible now?
Yes, ma'am, now it's clear.
Okay. Okay. Thank you. Thank you for letting me know. So yes, I think it has gone better, all the attendees. Yes, Brahmarambika, all the session recordings, you will be getting it, okay? And the recordings will be available to everyone, but we will need a few days to, to process the recording. So like within a week or so, you will be receiving the recording. All right. Yes. So as I was telling, so I will just repeat myself because of the voice problem. So in this session, we'll be talking about the applications of artificial intelligence in genomic, cancer genomic studies and research. And at the same time, we will also be looking at the future trends and also the limitations of AI in this particular field. So for that, I would like to welcome Dr. Nilofer here to the workshop. Welcome, ma'am, and thank you so much for taking out time from a Saturday afternoon and joining us here.
Yes, ma'am. Thank you, and very good afternoon to one and all. I hope I'm audible.
Yes, ma'am, you are.
Yes. So
Okay.
I just want to say thank you guys for joining. So yes, I am ready. So if Titi ma'am allows me, then we will proceed accordingly.
Yes, ma'am. Just 2 minutes. I
Yeah,
surely need
sure.
to introduce you to all the audience. So everybody who is joining here. So we have Dr. Nilofer Sheikh. Many of you might be already be knowing her because she is one of our oldest bio-IT specialists with Biotechnica. And she is very popular among all the students. They love their teaching, and I hope all of you will like her teaching today as well. So we are delighted to welcome Dr. Nilofer Sheikh, a bioinformatic expert with a PhD from MIT ADT University. She specializes in big data analysis of cancer omics and is skilled in AI, machine learning, and multi-omics integration. With several international publications and a granted Indian patent, Dr. Sheikh has made impactful contributions to precision cancer medicine and computational research. So there can be no other person more suitable for today's session apart from Dr. Sheikh. So we are eagerly waiting for your session, ma'am, and all the students. I want you to to be very much attentive during the class, and please note down your question, and you can ask ma'am during the QA session. Over to you, ma'am.
Okay. Thank you, Titi ma'am, for the kind overview. Highly appreciate. And I will take from now ahead with respect to the post-lunch session. So I hope that all of you are excited and all of you are ready for the same. Yes, guys? All of you are ready? Shall we begin? Great. Thank you. So I will first share my screen and then I will just give you a quick overview and then we will just proceed accordingly. So just let me know my screen is visible. I'm just sharing 1 second. Yeah. Yes, over there. It's visible, guys?
Yes, ma'am, it is visible.
Okay, ma'am, thank you. So as you all know that even Titi ma'am have given an overview that the session is entitled with Artificial Intelligence in Cancer Research, the data-driven and revolutionizing. Okay, so basically in this particular presentation, we are going to walk through a quick overview regarding introduction to artificial intelligence and machine learning in oncology. Followed with that, we are going to have an overview regarding how the artificial intelligence is playing a very significant role with the cancer predictions, prognosis, and even response for the same. Followed with that, we are even going to have an overview regarding what are the quick applications of this particular research area, okay, and how it is helping with respect to the emerging fields with the integration of multi-omics data. And last but not the least, we are going to see what are the challenges with respect to the big data and how we will get a future opportunity so that all of you can move one step ahead in your career to make a proper roadmap and achieve your research goals or as the career goals accordingly. Okay. So I hope that all of you get an overview, okay? Because now we are going to walk through with the most relevant and important overviews, which is I'm going to cover in the presentation. Followed with the presentation, today I'm also going to walk through, guys, with a quick demo, okay, about how the machine learning algorithms and even the deep learning algorithms, definitely deep learning part I'm not going to cover because it will take some certain time. But yeah, for sure, I will take a machine learning algorithm that to show you guys how we can build a model, okay? How we can use the biological textual data, which contains certain features of oncological targets, and then we can build a model, okay? So without further ado, I'm going to proceed. As you all can see that the first slide, which is get entitled with Artificial Intelligence in Cancer Research, the data-driven revolutionizing, okay? So basically revolutionizing, why? Because in this era, we are working with respect to the high throughput techniques and the tools, right? So as we are using the big data continuously, so there is really a need to understand this overview, okay? Even though, guys, you're coming from a different domain area, maybe you are corely having your expertise lies with the experimental techniques, right? Handling the overall experimental stuff, or else you are aware with some life science kind of domain where you know certain biological mechanisms and its targets. But now, as the era is moving with the big data analysis and we are looking for the bioinformatics kind of pipelines, right? So bioinformatics is one of the domain area where it is helping in the form of integrated way, okay? Because bioinformatics itself is an integration kind of field, okay? Where the biology, computer science, mathematical models, and even the predictive analysis, what we are able to achieve, it's based on that kind of integrated manner. And as the AI is getting again integrated with that stuff, so ultimately, it is making a revolutionizing kind of approaches with a data-driven kind of findings, right? And here, specifically in today's workshop, we are going to speak about oncological data or as the biological data accordingly. So why to discuss this? If that is your question, guys, so the quick answer is that it is transforming the future of cancer treatments or as the biological treatments through the intelligence data analysis and even the predictive modeling. How we can do that? If that is your question, then the first thing you guys have to really get an answer that what is artificial intelligence, right? So I hope that all of you are aware about AI, because nowadays artificial intelligence is playing a role in every area. It is not about only with biological stuff, but in every area we can see that the biology is like the AI is getting integrated, right, guys? So can I expect some answers like what is artificial intelligence? A quick answers, guesses, guys. What do you say? How you define artificial intelligence? Maybe in one word. Can I expect? I can see that it's like a very huge number of participants we have today, and I just really appreciate, guys, to all of you. Thank you for joining. So I can see that the answers are coming accordingly. So artificial intelligence is mimicking the human intelligence. Fantastic. That is really true. Training the machines to think like human. Wow. Okay, again a very, very good answer. So next I can see that it's a computer science that focus on creating the systems and machines. Fantastic. So I think the keywords which has been used in the answers is exactly the same thing which we are really looking for. And we can see, okay, we have the robotics, we have the machines which is get built up in very smarter way. Okay, and we are able to observe that in our daily routine life also. True. So it is a human-like intelligence which we are trying to integrate so that we can use for human applications, where it can be used for the proper analysis and even the helping hand kind of stuff in very simple way. Okay. So artificial intelligence is like right now is in the form of like helping hand. Maybe if you're using for any kind of perspective, or else you're using for the research and development in the large or the big pharmaceutical or as the industrial kind of way. Got it. So that is really true, guys. And thank you very much for giving a quick answers for the same. So if we talk about like very specific that what is artificial intelligence, machine learning, okay, and definitely followed with deep learning. So it is ultimately that artificial intelligence enables machines to mimic human intelligence, okay? And that is really true because all of you have given that kind of relevant answers for the same. So we are trying that artificial intelligence is a such kind of algorithm or as a model, you can say in the simple way, so that it can help us to make the process in a more simple and even in more accurate way. These are the two important things, guys, remember. Okay. So once you want to call your model is the artificial intelligence, okay, means your data has get trained till that particular point where it get like expert, or else the accuracy is perfect. Okay. So I used to take this kind of like universal example because this fit like this kind of example fit very perfect. Okay. So I hope that all of you definitely watch the robot movie, right? So there we saw that there were two robots in that movie, Chitty and the another robot, right? Where the villain has with them. Okay. So we saw that how the chittis get trained by providing multiple kind of tasks, okay, and then making expert and get an accurate in a such a level that the model is called artificial intelligence. And we saw that after getting trained like to years, okay, maybe like we saw that the Rajnikant, right? He used to like train the particular robot with like many years. And once he got a confident, like, yeah, the accuracy has reached the level, then that kind of robotics model, okay, one just for an example, we used to call as the artificial intelligence model. Smart. It is a smart model and it is an accurate. Two things, whenever you get achieve in your hand, guys, then only you will call your model as artificial intelligence model. Okay. So why I'm just concentrating this kind of stuff? Because if you observe that, as I mentioned, there was one more robot in that movie, okay? But, and that guy, the villain is asking about to give the gun, but the robot has provided bun, okay? So that is exactly what is not required, okay? But due to the similar synonyms, okay, the data is not get trained properly. And if that get occurs, guys, in the real world scenario, then you can just think about what will be the negative impact, right? And that should be get properly like trained, analyze, okay, cross-validate, and even get an accuracy. So that is exactly what we need. Okay, because artificial intelligence is all about accuracy. Okay, I hope that this quick example give you a clarity to understand that particular AI. Okay, make sense, guys? All of you are agree? Do this example suits better? Okay, because it's a quick example so that you will get a clarity accordingly. So why I have just taken that example? If that is our question, the answer is that we have two more models in our hand that is called machine learning. Okay, so why we are in the machine learning phase? So remember, guys, if you are a beginner, okay, if you are really eager to understand how to deep dive with artificial intelligence kind of domain area, then the first initial step is to start with machine learning. Okay, straightforward, machine learning. So to machine, you need to provide some data so that it can learn, and then it will try to give you some kind of output. So in machine learning, we are expecting the output should come up in the form of accuracy, means percentage. Like in this very simple way, guys, as we all know that during our school days, right? So we used to write the exam and then we used to get the results in the form of like percentage. So based on that, the college or else you can see that the class percentage will come. Okay, means it will depend on how many students get passed and how many got failed, right? So according to that also, there was like a thing we used to done like a class percentage we used to take place, right? Now all of you remember. So remember that AI is an upgraded thing only, okay, but now we are using in a very smarter way. Got it? The things were previously only, but we have not used in that level of thing, but now we are using that, we are polishing that, okay, and we are using in that kind of stuff, and that is still playing very impactful role dealing with today's kind of era, where the big data analysis, more predictive, okay, more quick of analysis we need, right? So that's why you can see that machine learning is something where we used to provide some data. That's why it is called as a data driven. Okay, so data driven algorithms to detect the hidden patterns and try to improve the predictions. So what basically we are doing, guys, remember, here in machine learning, we are continuously trying the data to learn in a such a way that it will reach that specific accuracy, maybe 70% of accuracy, okay, 80% of accuracy, or else 99% of accuracy. So it get varies based on the data set what you guys are providing. Okay, so the next part is about deep learning. So when and how I should use the deep learning? So once you think, guys, my machine is get trained very good, I have reached a significant accuracy with maybe a small stuff of data. Okay, so if I take one example, guys, over here for machine learning, maybe if you use the clinical data. Okay, so as we all know that we have get an experience of COVID-19, an outbreak, right? All of you. So we know that the outbreak was such a thing that we were need to have a significant drug, okay, which can treat that specific disease. So all were struggling, including the scientists, right? The experts in the field, the doctors, okay, and many of us have also struggled a lot. But there, if we just talk about how AI and this kind of machine learning has played a role, including bioinformatics hand in hand, it's nothing but it has provided to increase the accuracy by studying the data, right? To get to know the predictions out of that, okay? So we are able to get to know the specific vaccines for that, okay, the drug targets, because the data was stored in the bioinformatics platform. As we all know that the sequences, databases are there in bioinformatics platform, right? So we have pulled that sequence data, aligned against the viral target, and get to know the similarity based on the other organisms of our where they're in the databases. And based on that, we got to know that it's a viral target. These are some kind of cool, closely related viral targets are available, and which are the specific drugs has been given for that. So according to that, a comprehensive way, the biological mechanism is get to understood by taking help of bioinformatics data base, because the data driven is nothing but the complex databases are available where the large sequencing data, drug data, all is get stored in the proper platforms. Okay, so that's why I'm just point outing the thing that is bioinformatics is one of the field which help you guys to pull out the data. The data acquisition part is really important. So once we have a data, we will just study its pattern. Okay, we will apply the correct machine learning algorithms and check it out its accuracy. Once we think that the accuracy is reached with a significant level, as I've taken the COVID-19 example, right? So if I want to come up with like predictions, right, that the patient is alive or dead, okay, if that I want to get to know, then I need to study the different factors which is responsible for that kind of data. For example, I have taken a 1 lakh patient data. Remember, guys, okay? This is a very quick example so that again, all of you will get a clarity by working with machine learning. So if I have a 1 lakh patient data, irrespective towards a different geographical location, and I want to understand that how much patient is alive or dead, okay? So in that condition, I need to pull out some clinical data for that. Like what is the age of the patient, right? What is the disease status of the patient, okay? And do the patient has any kind of like a health disease, okay? What is its immune like responses and all that kind of stuff. So if the age is getting correlated with respect to the disease status, and based on this correlation, I'm able to understand that what will be the chances that the patient will alive or it will be get a death, okay? So that kind of predictions also you can do, guys. So how you're able to do that kind of predictions based on like having multiple factors or as the data sets in your hand. Or if you're able to make sense, okay? So that's why data is really important. With that data, you can proceed with ML part, okay? So again, the data should be in a such a way that we used to call as a supervised data, okay, in machine learning. Supervised data is nothing but that every column or the every feature should be relevant with the next important outcome. Like edge, we got to know that, right? All of you remember that during COVID-19, we used to see that that the edge is one of the important factor was there, okay, to say that the patient will be alive or dead. I'm agree. All of you agree with me. So I'm correct. So that we saw, right? So we used to get a continuous like kind of stuff that the edge is one of the factor responsible. And we saw that, we observed that in the real world, right? So that is the scenarios working with machine learning kind of stuff. So data is really important. That's why the patterns are nothing but within your data, how the correlations are there, and based on that, how the accuracy is there. So for example, if I'm moving one step ahead, like there will be like not 1 lakh data, I have a millions of data points, right? So in that condition, the deep learning algorithms will play a role, because deep learning depends on the neural network kind of approach, where we can analyze the complex data sets and with then we can study the features. Okay, without manual features is nothing but there is no need to manually pull out the features. Once you have provided the data in the curated format, okay, then the deep learning algorithm will work in the form of like neural network kind of concept. So what is neural network over here is very simple, like how our human brain works, right? So right now, if I just tell you guys that, can you give me an example, or can you tell me that in your like childhood, okay, any kind of memory, okay, you want to share, a very funny memory if you want to share, okay, if I tell you this thing, guys, okay, so what you will do? So your all of us, our brain will work in a very quick way, okay, so we will just go in the back and we will just recognize what was that memory, okay, which make me very happy, okay, the funny moment. And sometimes what happened that in our brain multiple pictures will come, right? All of you, the same experience you will get, right? What I used to get, right? Is that true? What do you say, guys? Is that true? Yeah. So if I tell you, please share me a memory, a happy moment in the past. So what you will do? You will start finding that memory and multiple pictures will come in your mind. Okay. And then one of the memory will get picked from that. Okay. And sometimes what happened on our face, a little smile also come, right? So we will just, sometimes we will just smile, and sometimes we used to say that is what exactly I want to say, the happy memory. Okay. So this is how our brain works. So in the similar way, deep learning will also work. It is one kind of mimicking the human intelligence in the form of neural network. So why neural? Because in our brain, there is a multiple neurons out there. Got it? And that's why we are able to pick the correct memory, correct data in the accurate way. Correct? All of you agree? That makes sense, right? What do you say? All of you? Fantastic, fantastic. Thank you. So that is a way where you guys have to actually correlate the things, because as I mentioned that AI and machine learning and deep learning is an upgraded version of all the things. Okay. So like statistical analysis, mathematical modeling, we used to do in our previous research also. In the past, many of the researches get published, right? And do you think that because of AI was not there, this kind of findings we didn't got? We got, okay, we got the novel findings, okay? We got the Nobel Prize of kind of findings, okay, based on like mathematical and statistical based modeling and predictions. So nowadays we are using in the smarter way, okay, and very what you can say is a accurate process way. That's why we are able to reach the predictions. Okay, as I mentioned earlier, that the predictions is the most important, and that is clearly we can see. Okay, I hope that all of you get an overview. Okay, so I will not take much time. I will proceed quickly because I want to share a quick demo also. So why AI is matters in cancer? If that is your question, guys, and today we are trying to answer that. So because you can see that in the coming year, the new cases is getting like increased by 20 million. All of you are getting my point, right? So by 2050, we are expecting around 20 million new cases. And it is the second leading cause of death globally. So it's really need to take care, guys, okay? So we, the upcoming researchers, you guys, the upcoming researchers has to really move one step ahead to just understand the stuff. Okay, and today you have take one step ahead. Okay, you have kickstart your career, guys. Okay, and I just highly appreciate to all of you in that context. So be in the track so that you can achieve your goals accordingly. Okay, so as you can see that we are very corely looking towards the specific area. Okay, and if that is the huge number of cases, you guys can see that. So it's really need to take care, right? So AI has accelerate not only predictions but also the earlier detection. Remember. So ultimately, artificial intelligence we are using in such a way that we can look for the earlier detection, precise diagnosis, and even the personalized treatments in future. And that is able to do based on the vast integration of imaging data, that is biomedical imaging, okay, genomics data and the clinical data, the example which I have taken in my previous slide. Okay, the predictions, how you will come up with the predictions by having a clinical data in your hand. Okay, and in bioinformatics, we used to do comprehensively because why? The databases are available. Straightforward answer for them. Okay, so I hope that it get clear. Moving to the next slide is that I hope my slides are moving, right? So the next is artificial intelligence powers in predictions model. Okay, means what? So if we take a specific data such as tumor progression analysis, so deep learning models will predict the specific progressions and even the patient survival by analyzing the biomedical image data. So what are those biomedical image data is nothing but the MRI data, okay, histopathological data, okay, CT scan data, and many more data sets. Okay, so that is how the biomedical images data will play a role. Molecular data is all about like your sequencing data, okay, next generation sequencing data, or as the gene expressions data, got it, proteomics quantitative and the qualitative data, so on. So we can even move ahead to classify the different types of oncological targets, because you know that, okay, today the only disease is not responsible, guys. If you just explore the different research paper, you can see that within the disease, there are different types and subtypes are playing role. Okay, like for breast cancer, if you take, there are HER2, luminal A, luminal B, okay, triple negative. So you can see that the triple negative, this kind of subtype is getting more resistance patterns irrespective towards different population or the cohort data. And exactly there, guys, you have to, okay, concentrate your research. Okay, you need to go in that specific area so that you can achieve the goals of precision medicine, okay, or as the personalized therapies to get to identify, because enough of research is done. Okay, and as I mentioned earlier, that in the smarter way we have to think to deep dive, okay, with the target oriented things, okay, and then take help of multi-omics approach to understand how we can look for the specific biomarker findings within that type, resistance patterns, and so on, right? The next thing is that immune response predictions also, because clinical data is one of the important factor. Okay, so if we are trying to work on the molecular findings, so how much the molecular finding is relevant to the clinical data is exactly what we need for the predictions. Okay, and for that, we are also having certain kind of models which help us to predict the immune response. Okay, by studying the immunotherapy optimizations, taking the AI data sets with the predictions and so on. Okay, so that is how we can do the predictive analysis. The next thing is that we can even move ahead with AI with multiple approaches. Just have a look, guys. Okay, it's very good reference image, okay, from the research paper, which tell you an overview about how artificial intelligence in the oncology is playing a role. Okay, it is playing with the two perspective, guys. The first is diagnostic point of view, and how after diagnosis, how you can move ahead for the personalized treatment or the therapeutic treatments accordingly. So diagnostic is nothing but like how we will diagnose the specific data in the form of the patient. Okay, so in that we have the microscope based research, okay, or the image based research. Okay, tumor screening, earlier detection. I think pre lunch session, you saw that, okay? Our speaker has given an overview that how we can take the liquid biospecific data, okay, to do the predictions with earlier kind of stuff. So this is the kind of stuff, okay, guys, you have to integrate. So then stages study, okay, tumor stages, staging, new therapeutic targets, how we can discover, okay, how we can look for the designing a new drug targets by using the drug repurposing kind of concepts, right? Clinical decisions, how we can make ahead with the clinical research kind of domain area, okay, and how the imaging, which I've explained in my previous slide, the biomedical images, how we can use. And this, all the things depends on this factors, guys, remember, okay? So the factor is that how we can reach to the survival significance, okay? How much we can say that the predictions depends on the survival significance, and that is what we need, okay? All of you are clear so far, whatever the points we have taken. Okay, it is highly correlating, I hope so. It making sense to all of you, right? Yeah, clear, all of you? It's aligning with the discussion. Okay, great, good to know. The next thing is that you can see that it's not about like only diagnostic and the treatment, but also AI is looking for like how the patient input data can be used. Okay, diagnosis can be done, proper therapeutic guidance can be get provided, treatment assistance, okay, because that is really important. Because if you just go and visit to the nutritions, okay, nutritions provide you the proper assessment, okay, it will give you a proper chart for the nutritions kind of stuff. So that is like a routine kind of stuff. If you want to go deeper insights with this kind of oncological kind of data sets, viral kind of data sets, okay, immunotherapy kind of data sets. So this kind of stuff also we can do with AI, okay. Then the predictions, okay, survivals, inputs data accordingly. The next thing is that the benefit of AI in oncology is that it's more accurate. I'm just repeating for my first slide itself, okay. So AI is about accuracy. So accurate diagnosis need to be done, streamlined workflows should be get designed, okay, standard pipelines, okay. Improve the patient's outcome should be taken care while working with AI, and that is we're able to see, right? So reduce the health cost. So I think we have discussed in the previous session, guys, okay, like the Tata Memorial hospitals are actually looking towards like this kind of stuff, right? Improve the patient outcomes, reduce the health cost. The government is sponsoring this kind of projects with AI predictions and that kind of stuff. So that is exactly where the AI is playing role. So if we just talk with respect to AI in drug discovery, so ultimately we are looking for the response predictions, like how we can do the drug combination screening. Because in my previous slide, I have given one a keyword, right? Remember, guys? It's all about like drug repurposing kind of concept, okay? Because now we have enough of drugs available in the market and also in the pharmaceutical research, okay? But do you think, guys, there is no need for the new drug? It is, okay? Even though like millions and billions of compounds and drugs are available, but still, okay, many of the researchers are in the race to identify a new and the potential drug targets. Agree all of you with me? Okay. And again, in the same way, I used to say that if you want to think in the smarter way, then we have an approach in our hand, which is called as a drug repurposing. So we have even published certain research papers on drug repurposing kind of concepts, guys. And I just really recommend all of you guys to please explore this kind of research paper. It's already there, okay? The only thing is that you need to explore that. So drug repurposing screening or the drug combination screening, where the machine learning will help us to perform a predictive analysis based on its drug bioactivity property, like a Lipinskill property, I hope you all of you know, admit property, right? So that's why we used to see that how the drug is potent, and that's why you can even see that sometimes the drug is get released into the market. Some of the drug will even get withdrawn. Why? Okay, because some toxic effects get like observed in the population-based study. So this kind of things used to be done by the withdrawing the drug from the market. Okay, and if it is that done, then you can see, guys, that how much time it is get spent on that for screening, clinical analysis, okay, post marketing, okay, and then releasing that kind of drugs. So then again, withdrawing it is really a challenging stuff. That's why we are taking help of AI and ML, bioinformatics kind of stuff. Okay. And then toxicity predictions and even the personalized treatments, it's all interlinked with one another. Okay. That's why this is how the drug discovery developments has lead towards identification of drug target. That is how the AI will help you. That how the drug repurposing kind of done based on the existing drug. So drug repurposing is something like repurposing the drug from the existing drug information, okay, like screening, correct, based on its a functional group and the parent compound. So molecular simulations we used to do in the comprehensive way. So we used to do that kind of stuff in the cloud computing, and that's the kind of stuff. Clinical decision supports help us to do more predictive analysis, right? And that's why the tumor image analysis also we are able to reach. So it is not about only with working text data. Remember, guys, okay? AI has also reached to work with image data sets of biomedical targets. So again, again one more example I want to take is for the clinical, like the image analysis is for the COVID-19. Okay, so you will see that lots of projects or the research paper get published on the lung images. Okay, and then they have done the automated kind of detections. Okay, because nowadays we are not only using AI, but nowadays we are using the generative AI. So generative AI is a concept where the model, pre-trained models get stored, and we are using directly that. Okay, and in that stuff, we are making an automations kind of production, predictions and even the detection. FDA approved tools we are using so that it will assist in the form of like robotics, okay, predictions in the speed up manner. Generative AI, that is exactly what I've mentioned, okay, which makes us the high quality of predictions irrespective towards the data sets you are using, okay. So in that stuff, we are able to get the earlier detections, okay. We are able to do the multi-modeling kind of screening, guys, remember, okay. So what is mean by multi-model is nothing but using more than one models, okay, so that we will get a more accuracy with very insightful finding, okay. And liquid biopsy integration, I think that is getting related again, and because it is making a predictions, okay, where we are able to do the predictions based on data and even the image improvement, okay, how the diagnostic can be done in the earlier stages. And that makes a risk predictions also. Remember, guys, okay? Again, really important thing that why we are doing this earlier detection, why we are achieving, because the earlier detection risk predictions can be done. And this we are able to achieve based on like multi-model screening, artificial intelligence kind of stuff. Make sense? Okay. I hope that it is correlating, right? All of you are clear. Got it. So then multi-omics integration is all about like where we are taking help of genomics data. Okay, I hope you all of you know genomics data. Transcriptomics data sets, gene expressions pattern. Metabolomics data is also important because ultimately we are looking towards the drug targets, right? So we need to understand the drug pathways, mechanisms, and kind of stuff, right? Then the epigenomics, where the methylations analysis, proteomics, where the protein quantification study can be done, so that we will get the accuracy with the finding, okay? So AI has integrated models with the complex data sets where we are able to classify the disease type, predictions, okay, in the holistic way, okay, to understand the overall mechanism and even the therapy. Got it? So that's why the breakthrough and the examples are there, guys. You can see many examples are there, like a neural forest model, okay, like as I mentioned, it like based on the neural network, it will work, which will work on the deep fixable neural forecast model. Okay, like encoders are there to encode our data, decoders are there will decode your data. Means encoding is that once you input the data, it will start converting into the computer readable format, like a binary. Okay, that is called as an encoding. And then it will again decode the data so that the human can read the data, do the interpretations, analysis. Okay, so trivial microenvironments kind of study also you can achieve based on AI to get the immune therapy response. Biomarker discovery is the ultimate goal where the AI and the oncological stuff is playing integrated role. Okay. So the key challenges in AI oncology is ethical point of view, because I saw that in the previous pre-lunch session. Okay, one of the students has asked the question like, what will be the disadvantage or is the negative use of genomics, right? So in that context, the ethical protocols are there. Okay, the ethical guidelines are there, so that really need to take care so that the challenges will be get overcome, so that we can understand the AI predictions, integrations with a certain kind of clinical data, oncological data, because this is a very controlled data, okay, the research data. So how the complex interpretability can be done, okay, how the clinical validations in the safer point and the regulatory approvals kind of pipelines or as the protocols can be get studied. So that is also a things which is really need to take care, okay. Challenges are nothing but where we are looking for the limitations of AI, ML in biology, and even the bioinformatics. So remember, guys, this is my personal experience, and I think every researchers will definitely face this experience while working with artificial intelligence, machine learning kind of stuff. It's nothing but the data collection or data acquisition part. It's a part of research, guys. So never think that I am facing a challenge while working on the research data. I'm not able to get the data. Okay, it's a part of research, guys. It's okay. So it's really fine that if you are going with this kind of challenge, the challenge can overcome if you stick with or refer the proper databases, that is bioinformatics platform. Algorithm design is nothing but how you will use of machine learning to build your model, deep learning to get a neural network kind of concept, AI to build your accuracy kind of model. Okay, so this is ultimately unlocking the potential of AI and machine learning technology to overcome the obstacles and explore the field of biology and even the bioinformatics. Okay. Next, we have the addressing the challenges, right? So challenges are nothing but the privacy, okay? The data privacy, okay, how we can do the proper security, okay? So even in that kind of stuff, AI is playing a role. How the explainable AI is nothing but improve the accuracy and the clinical expectance can be done. Clinical validation is really important because whatever we are predicting, okay, nowadays you can see, guys, AI is not only predicting, but we are reaching towards the validations also. Okay, many companies are there who are taking the AI pipelines and they are working on R&D and even going for the clinical validations to deal with real world evidences kind of stuff. Okay, so they are overcoming that kind of challenge and problem with the clinical validations. So that is what the AI is doing right now. It is not predicting, but it is also going one step ahead for the validations. So that's why the future advancement is that. So it is getting integrated with genetics data, genomics research, many what you can say as a comprehensive kind of data sets such as oncology, neurological, clinical study, okay, where the AI applications is getting an advancements where unlocking the mystery of biology. Okay, remember guys, this is very important that AI and multiomics is unlocking the mystery within the biological world. And that is what we need, okay, considering the smarter world, okay, where we are using the smarter technology and that kind of stuff. Okay, so the overall application of AI in healthcare is that patient care, medical imaging, because you see that we have the robotics, okay, surgical robots are there, okay, with the physicians kind of application. Research kind of domain is that we are able to pull out the data, do the modeling for drug discovery, drug designing, and that kind of stuff. I hope that it get a clear picture now, all of you, right? Once we are talking about AI applications in healthcare. So think in this way, guys, patient care, how you can look for the automated assistance, real-time monitoring, okay, and even looking for like medical imaging with the diagnostic kind of stuff. Physicians kind of applications is nothing but robotics, okay, clinical trials has been used, and research and development is all about like pulling the data, analyzing, and making predictions. Okay, agree? So the overall conclusion over here is that AI is making a transformative promise in oncology. Okay, if we just talk about with oncology. If we just talk about with biology, it's again playing a role with reshaping the treatments, okay, how we are making the proper management, diagnosis, and even coming with unpredictable precisions accordingly. So ultimately, it's reshaping, guys, okay? It is playing a global impact, remember, okay? So the overall application is the global impact, okay? It is continuously making innovations and getting integrated where the improvement in the patient outcome globally can be done. That's why the different sponsored projects is getting lead. Okay, that's why the different startups are coming into the picture. Okay, and even the government is leading towards that. So that's why data-driven future is awaiting, guys, for all of you. Okay, and that's why the future is all about data, AI, how the power of AI can be used, and that is how it will lead you with kind of outcomes, guys, predictive outcomes, more accurate outcomes. So that is all about from my side with the theoretical overview and even the conceptual overview for my side. Okay, I do have 10 minutes with me. Okay, I want to take this 10 minutes to walk through, guys, with a quick demo. So are you guys are ready and excited for that? Okay. Can you please let me know the overview with the conceptual and the theoretical which we have discussed? It's clear to all of you. I just try to make very simple, but the key points whichever we saw on the screen, okay, that is exactly, guys, which you need to capture. Okay, that's why purposely that slides contain the relevant keywords and the approaches. So if you even stick with that approach also and then deep dive your research by research survey, literature survey, working with bioinformatics databases tools, and definitely the guidance with all that kind of integrated stuff where the Biotechnica is a one-stop solution for all of you, okay? Because Biotechnica is not only providing the workshops and this kind of overview, but also we have a separate courses for that, okay? Where we used to guide the upcoming students and the researchers like you to understand how those integrations can be done, okay? So I think that is how you can definitely take that opportunity and think in that kind of perspective. Make sense, guys? All of you agree with me? What do you say? I will not take much time. I will just proceed. So I have stopped sharing the screen. Okay. And please allow me, guys, to show you one example. All of you ready? Shall I? What do you say, guys? All of you ready? Great. So quickly. Definitely, I know that many of you have many questions. Definitely, we'll try to answer that. As Siti ma'am has mentioned that we have a separate QA session accordingly. But I think right now we need to like take the overview, right? What do you say, right? So this is one example. I really want to show you guys that how artificial intelligence and machine learning kind of stuff is using with the real-time data and working with this kind of oncological data, biological data of your interest, right? Sorry. So all of you are able to see the screen? So what the screen tell you guys, the example? Okay, I hope that all of you are able to see the example on the screen, right? This data is about lung cancer. Okay, simple, quick. Okay, so lung cancer predictions, how we can do using machine learning. So if that is your thought, guys, okay, if that is what you're looking for, then this is one of the example will be. Okay, so how the predictions can be done and how you will achieve that goals with the predictions and the accuracy by taking help of biological data, which is get pulled out from the bioinformatics databases. And the next thing is that taking help of machine learning algorithms to build a model. And for that particular purpose, remember guys, one more skills what you need to upgrade is programming. Okay, definitely not in an expert way, but if you think you are even a beginner in the programming also, it's more than enough. Okay, again I want to repeat. So if you think you are a beginner, okay, very new towards the programming also, it's more than enough. Okay, because once you just start and being in that kind of loop, right, automatically that skills will get upgraded. Okay, so that's why you can see that here the example which is going to get processed using the programming language called Python. Okay, but again I want to tell you that guys, the environment what we need to this process this kind of data, for that we need one kind of editor or else the environment we used to call. So right now on the screen what you guys can see that it's a cloud based environment. So what is now cloud? Cloud is nothing but like on web we are processing this data. Can you see that guys? Smart, right? So we have to be smart. So here the thing is that there is no need to process this data on your system, on your laptop accordingly. No worries, okay? So we have a smart editors right now available where we can process this kind of oncological data, biological data on cloud. So for that we have Google Cloud and for that we have Google Colab, okay? So Google Colab is nothing but an environment where the processing of your data can be done. Okay, so this is the environment guys, where you can see that it is get connected with your Gmail account. Okay, you have to log in with that, and you can see that over here Gemini is there, smart editor. Okay, and then you can get connected with the system. Okay, basically why I have mentioned as a cloud, because here it will give you a space. Okay, so you can see that RAM and disk memory. Can you see that all of you? It's providing you around 107.272 GB disk memory. Okay, where you can process your data. Okay, smart, right? So take help of this kind of cloud services where it's a free guys. Again, I want to tell you, it's a free. Okay, so you can try this. You can just log in with your Gmail account and just use this editor and then launch this example. So the quick TOC, that is a table of content in this example, is that you will get an installation of the library. Definitely, I'm not going to deep dive with that, but this is important. Library is nothing but the pipeline, the packages need to install so that you will understand how to process that data. A very simple example I want to tell you guys, like libraries are nothing but like calling the packages. Okay, like how you guys have registered for this workshop, right? So once you have registered, then today you have joined. Okay, why? To gain this overall knowledge. So in the same way, libraries are something like that where you need to install, you need to do the registration of that relevant packages so that the data will get processed. Because computer is all about like providing certain kind of algorithm to understand what need to be done. Got it? So that is how the data libraries get import, data sets get import. EDA is called as exploratory data analysis, data visualization, so that you will understand the insights, and definitely the last is building a model. Okay, so I will quickly walk through guys with an overview. Definitely, I'm not going able to take a comprehensive discussion, but the point will be like to show you guys that how this study focus on the earlier stage detection. Can you see that? Okay. So what we have discussed in the presentation, the same thing you guys can get reflect on the practical stuff. Okay. And it's possible. And today we are able to do that. Earlier stage detections can be done. How? By taking help of like a genetic k-near neighbor kind of algorithms. Okay. Or else taking help of like a k-near neighbor algorithms by even using image data, text data sets of your interest. Okay. That is the thing we can do. So the model which is being used over here, have a look guys, this is important. Models are nothing but machine learning models. So in that we have the models in the form of like support vector machine, random forest, k-neighbor classifier, ANN, artificial neural network, okay, voting classifier, stacking classifier, logistic regression, decision tree. So if you just explore guys, you will get lots of reference for that, okay. And that is how we have the algorithms with us. Then importing the libraries are nothing but like NumPy. Okay, you can see that NumPy talks about numerical data. So tell your environment that I'm working on the numerical data. So for that, you need to install the NumPy. Okay, pandas to frame your data in the tabular format. Okay, Seaborn and Matplotlib, the package name itself tell you, okay, this is a smart editor. You can see, guys. So just by moving my cursor, I'm able to see the information. All of you agree with me? Okay, so it's telling me that Matplotlib is all about work interactively, which give you interactive support or else give you a visualization, interactive, correct? So where you can save the PDF, SVG, PNG images accordingly. Correct, all of you? Clear? Okay. Plotly is again an interactive package. And then scikit-learn is for machine learning pre-processing and splitting the data in the form of training and testing. You can see that. Okay, it's smart editor. Got it. So as I mentioned that you need to give the data in the form of binary, encoding that. Remember, guys, in the presentation, I have mentioned that, right? And then we need to provide the data set. Okay, the data is get provided. And you have to process this data, guys. Okay, the only point over here is to show you that how you can process the data. Actual data set is available over here, that the table of content, okay, introduction, then the algorithms which is used, then importing the replica library, importing the data sets, performing the EDA. Just have a look, guys, this is important. So the data which is get used over here is a gender. Remember, I have taken this data. Okay, I will take only 5 minutes, guys, then we can wind up. Gender, age, smoking factor, okay, yellow finger, okay. So remember, guys, this overall columns are nothing but the features. So in machine learning, we used to call as the features, like how the one column is responsible towards the next. So if I want to tell the particular, see, lung cancer. So yes or no, that the patient is having the disease or not, on the basis of what I used to say, only by telling that the age is responsible, do you think, guys, we can say that it is a positive or negative? Do you think, guys, it make even the scientific sense on that, or else do you think that is a correct way of predicting? Only by telling that age, we can say no. Even you can see that this kind of factors are responsible, and that factors have some correlation. Means if the age is factor, do the smoking is also getting altered? Do the gender is also responsible? Okay. Do the other kind of alcoholic consumption, coughing, chest pain is also responsible? Okay, if all the things are correlating one after the another, then the chances of predictions will be get higher. Make sense, guys? All of you are clear? Okay. Guys, all of you are clear? It's making sense, right? Don't think like very deeper. Okay, just think in the overview kind of conceptual way. Okay, because as I mentioned in my presentation, that the practical way and doing the actual implementation, we have separate courses available. So no worry for that. Okay, just think in the kind of conceptual way. Okay, so these are the factors responsible. So this is called as a data framing, frame in the tabular format. Okay, then describe the data. Is there any null value? This is called as a pre-processing step. Okay, there should be no null, right? Then data visualization, like edge, based on the edge, you can make this kind of interactive plot. Can you see, guys, all of you? This beautiful analysis you can do within Python. Okay, you can see, have a look. I can select this. See, okay, I can select this kind of data which I need to pre-present. So this kind of interactive analysis can be done. Can you see that, all of you? A heat map. This heat maps can be used directly for the publication quality figures. Okay, that is the beauty of programming and even the bioinformatics stuff. Just have a look, guys, the patient of lung cancer, yes or no. This is the count, right? So then yellow finger is there. So what will be the yes or no kind of stuff is done in the form of data sets. Okay, so likewise, the overall EDA analysis is done. Okay, now if you want to tell that the gender is responsible, or as the if the disease is there, if that is what you want to find, then what you can do, guys, you can encode this data, which I've explained in my presentation. Okay, gender for male is 1 and for female is 0. Okay, and the disease is 1. If the disease is there, yes, then make a 1. And if the disease is not there, so make 0. Okay, so in this way, what happened, guys, you will have a quick predictions. Okay, yes, no, positive, negative. Okay, alive, death. So this will be a quick prediction, and that is a smarter thing what machine learning is doing. Now makes sense, all of you, right? So in that way, gender and the disease status is given, and you can see now, whenever the gender is coming, okay, male 1, female 0 is coming, okay, and likewise the status, you can see that has also come 1 and 0, where the patient is detect yes or no, right? So if that is our data, then how we can build to look for the feature? Can you see that, all of you? Feature selection and correlation. Then according to that, the models will start. Okay, random forest model, k-near model. Okay, so one of the model which is used over here is the SVM, that is support vector machine. Okay, guys, every algorithm will have that significant principle. Okay, sklearn, accuracy. Okay, so you can see that 97% of accuracy we are able to reach, or 96. Okay, why? Because the data was correct, and we got to know the proper correlation, and then based on that, the accuracy is nothing but the percentage of accuracy. Okay, predictions we are able to know. Now you're getting clarity, guys. Okay, so I will take another 1 minute. I will just wind up the session. Okay, sorry for like quickly wrapping the things, but we need to stick with our time. Okay, but I hope that what I want to provide an overview. Okay, I hope that is getting reached to all of you guys, right? So having this kind of data, we are able to reach the accuracy like 96%, 97% of accuracy we are able to reach. Like in that, we have a random forest, which give again the model like 96%. Okay, so like this kind of comparative analysis we can perform, and then we can even move ahead to get the modeling. Okay, so this we used to do in the form of like the accuracy, and I will just show you guys. Okay, the accuracy comparisons can be even done, like how the decision tree is giving accuracy, logistic regressions, ANN model, SVM model. Okay, and then you can come up with the beautiful representations of your accuracy model. Can you see that comparison of models, accuracy, precision, how much precision it is? Okay, recall values accordingly. So the takeaway point which is given by this particular author is that this will give you predictions to work on the multilinear regressions kind of modeling, comparison, and providing you the accuracy if you take your data and this kind of stuff. Make sense, guys, all of you? Did you get an overview? So I will just stop sharing my screen without further ado. I hope that all of you get my point of view with a theoretical conceptual discussion and a quick practical overview. Okay, all of you are able to correlate both the things? Yes, guys, what do you say? Do it make sense? Clear? Great. Thank you guys for the quick feedbacks. So I will just stop from my end. Yes, ma'am, I'm done from my end.
Thank you so much, Dr. Nilofer. What insightful session you have taken. Like, it's a power-packed session it was. You gave the idea of theory. You even gave a quick demonstration with the demo. That was so much wonderful. Thank you so much, ma'am.
Thank you, ma'am. Thank you very much.
All right. So I guess, yes, I can see the messages from all the attendees. And yes, I am all equally excited. Like, you people have also given us such a good review, right? The session was power-packed, isn't it? Okay, so I don't think anybody is feeling sleepy. Of course not, right? After such a wonderful session. No. Okay. Not sleepy at all. Great. Awesome. So with that, we are ready to move on to our next session. And our next session, as you know, will be the career guidance session in this particular field of cancer genomics. So cancer genomics, as you know, many things have changed since last decades. Okay, the way the research is done, the way the things are looked at, the way we have tried to develop new kind of therapies, and like this field is so much so much flexible and versatile that it is changing every single second that we are talking about. So we are going to move on, and with that I would like to welcome
Yes, am I audible? Good afternoon, everyone. Good afternoon, ma'am. Thank you, and I will take up from here.
Yeah, just 2 minutes, ma'am. So
Yeah,
as you
sure.
guys know, this session will be about career guidance, and Samrita ma'am is totally ready to guide you. What all are the steps that you need to take, and how you have to plan your career in this particular field. Before we start, let me give you a brief introduction about Samrita ma'am. So Samrita ma'am, she holds a MSc and BA along with her NET qualification and has been actively involved in academics for over 11 years, specializing in cell biology, genetics, and plant biotechnology. She is a master's from Calcutta University and has begun her teaching career as a lecturer at the college level, where she served for 5 years before joining Biotechnica. She has also served as an instructor in a UGC-sponsored national workshop on mushrooms cultivation. Her professional philosophy is deeply rooted in the belief that gathering knowledge is a passion and spreading it is a devotion. So with that, we are waiting eagerly for your session, ma'am. So over to you.
Thank you, ma'am. So I'll just share my screen. I hope the screen is visible.
Yes, ma'am, it is.
Okay. So today's workshop, from whatever sessions you have listened to from the morning, I'm sure you have got a very deep insight into what is happening currently, right? How the research is being conducted and what is the current trend. And as you can see, this is a very promising field, which is evolving. And in future, this is really going to open up a lot of opportunities. So you should be ready for the future, right? You should be able to grab all the new things which are coming up and also know how to implement that in your career, right? So in this session, I will be talking about what you can do and give you things in a very summarized way, like what is happening, right? What are the key features there are in different fields? Because as you can see here, there are three fields which are collaborating together. There is cancer genomics, there is clinical research, and there is AI. If you look at these three fields separately, then separately you can master yourself in each of these three fields. But when it comes to implementation, when it comes to work, right, all these three fields can be coordinated, and that really speeds up the diagnosis, and that really gives us very good results, very personalized treatments, right? So it gives us a promise that yes, we can survive cancer. We can, maybe in the future, we can outcompete cancer as well. Okay. So when talking about career, of course, the first thing that comes to our mind is academic, right? So this has been the trend so far. We have always thought in that line that, okay, we are going to study. We're going to do the BSc, MSc, then do the PhD, right? So that is a fundamental step. That is a fundamental uh, path that will always be there, right? Because you ultimately have to do research in order to understand how the things are working. If you don't know how cancer is working, how it is changing, how mutagenesis is happening, how metastasis is happening, what if it has taken up any new path, if new genes has been is getting altered, which is which is leading to cancer. If you don't know these fundamentals, you will not be able to generate the idea. You won't be able to understand what is happening. This is the baseline, right? So this is research scientists roles are always going to be open for you. So all of you who are interested in research, who are want to dig deep into the field of cancer, what is happening, how it is happening, go for the molecular level, cellular level analysis, transcriptomic analysis, and all. Academia, this is the field for you. Okay, so here you can be the research scientist, you can be the principal investigator. After that, you can be the professor and can have your own team to conduct research, right? Go for postdocs. Obviously, this will include a lot of like hypothesis-driven research, publications so as well, because see, whatever you are like whatever results you are getting from your experiment. Okay, so first and foremost, you have to get, develop some idea, that you have to develop some kind of hypothesis, right? And if your hypothesis stands true, okay, so you have to like communicate that idea, that result to the research, to the scientific world. Okay, because like you, there will be many others who are working on this. So as you know that there are many institutes which are working on this, like almost all the institutes have a cancer genomic lab, and there are government and university funding and grants are there, right? So whoever is interested in core science, you can take up the research path. Okay. So for that, obviously, first develop a very good concept when you are studying as a student during your BScs or MScs or MTechs. Okay. And then join the lab for more, exploration. However, there is the BTech industry also. So those who wants to like go into the industry, okay, you have done your BTechs or MTechs and you want to get into the industry. Of course, there is industry which is hiring candidates, right? And their main role will be like as genomics analyst, molecular biologist, research associates, okay? And these companies, they are also in some kind of research. Okay, they are like dealing with these companies on one end deals with some core research, but on the other side, they also deal with some data, the real-time data which are coming up, okay, from the field. So field data, you can say. So Illumina, Thermo Fisher's, genomic startups, they are all there, okay? And their main focus lies in the part of translating the discoveries into diagnostic and tools. So what you study as a researcher, what you discover, if it is applicable or not, right, in the larger scale, that part is taken up by the biotech industry. Okay, they see how the lab work can be converted into reality. Okay. Then, of course, whenever it comes to cancer or any kind of disease, pharma industry plays a very, very important role. Okay, so there's a wide pharma industry out there, which is doing an incredible job of discovering new drugs, of designing new drugs which are more precise, which are having less impacts. I mean, side impacts, right? And they are very targeted because that is what is more needed. More precision is what is needed. So in the pharma industry also, you will have the research, R&D. Okay, that is the core. Again, as I said, that is a fundamental. Research work, R&D work is a fundamental, whether you do it in a university or whether you do it in some kind of company, right? In a particular company. And from there, you again generate some real-time data, some data that you get from the patients, you get from the doctors, right? And from those data, you understand what is the troubleshoot, right? What you have to address and what kind of medicine is required. So pharma industry is the one which is looking into that part, okay? So this is one industry which is involved in clinical data. They are looking not only for researchers, but they are also looking for the clinical data scientists translational scientists, and drug development. So clinical data scientists are the ones who are actually going to understand the real time impact of the drug. Okay, they are going to go up to the people, to the patients, to the doctors, and understand if the drug that has been introduced, that has been newly formed, whether that is effective or not. What is the percentage? What is the efficacy of it? And translational scientists, they are the one who are actually, if the drug is discovered, then they are the one which is like introducing it to the market. Okay, and they're going to see how this drug works and all. And of course, there is drug development. So these things are again collaborated together. Okay, they are not uh, independent, but they are like dependent upon each other. So there are different departments, R&D, precision medicine, and pharmacogenomics. Okay. And here these industries, they mainly target in understanding the target identification, the companion diagnostics. Okay. Now, when I talk about these industries, as I have said, clinical research is again it is a part of the pharma industry, but again you can take up this sector as a separate industry as well. Okay, clinical research, there's a huge demand nowadays for clinical research because there's a lot of data that has been generated. Okay, so we need specialists who can analyze those data, who can who can tell us what kind of solution is needed. Okay, what can be done. So that is a separate field which is evolving right now, clinical research. So what is the role of those who are involved in this clinical research? Where they have the role of clinical research associate, they can be a clinical trial coordinator, or they can be the data managers. Okay, excuse me. Now, there are several organizations, okay, like CROs, IQ, VIA, Parallax, hospitals. Hospitals are the place where you get a lot of data, right? Cancer centers, okay, again, you can get data from these organizations. And what is the focus of this particular domain is regulatory trials, patient data analysis, FDA, EMA protocols. Okay. So you have to also limit yourself to the ethical aspect of clinical research. You need to know that as well. And as I said, that there's a lot of data that has been generated over the few years. Okay, so you need to analyze those data. And who are going to analyze that data? Obviously, those who has the concept of data analysis, right? So this is again another domain. The AI startups are there. Lots of AI startups are there who are hiring these the candidates who has knowledge in data analysis, right? And machine learning. And as ma'am has already shown you, all the AI, all the data analysis concepts that you need to know. So this sector mainly is looking for machine learning engineer, data scientist, computational oncologist. Okay, who are not going to go into the lab, perform the experiment. No, not like that. They're not going to go and to the hospitals and collect the data. No, but they are rather going to analyze the data which they are receiving. Okay, and from there they are going to produce some predictive modeling. Cancer, they're going to suggest some cancer diagnostic. And yes, AI has also got the capacity to analyze images. AI is developed. Okay, initially AI didn't have this capacity. AI could only understand the language, right? You type the sentences, AI understood the message that you could get. It could only give the output in the data as well, okay, in the sentence wise and numeric wise. But now there are applications which has been formed, which has been created, which can analyze data, images. So suppose if you give a image of a cross section of a tumor, okay, you give the image of a cross section of a tumor, or and you give it to AI and you ask the AI like what kind of cancer it is developing, or does it have a possibility of developing cancer, or like which other cells which are like triggering the cancer formation, okay, and many other questions. If you try to gather that, the AI will be able to tell you all those answers. Okay, so it uses, it has the capacity of that. So understand, like if you look at a picture, all right, it might take you months to get some information out of it, but AI can just do it very quickly and give you the information. And it has been trialed and tested as well that the information AI is giving is accurate. Okay, at least 99% accurate data it is giving. And it is being utilized also nowadays. So if you are thinking like whether you should go for this cancer research or not, then let me tell you that over the years, if I take the data, see, cancer research, okay, the demand, okay, or the activity or the work, you can say, it has increased and it is increasing. Can you see? This is the data from 2021, right? And there are two data giving here: cancer diagnostics and cancer treatment. Okay. And with AI evolving, it is expected that this field is going to increase. There's going to be more of cancer diagnostics and better cancer treatments. Okay, so lot of research will be funded based on cancer treatments. On cancer treatments based on the cancer diagnostics, because now you are being able to rapidly analyze the data and also formulate the solution. Okay, at least a probable solution. Okay, which obviously will be trial and tested. Now, cancer research, if you see, there are 4 types: basic research, translational research, clinical research, and population research. So basic research is the typical R&D work, right? It is absolutely based on the results. So you go for the cellular study, molecular study, genetics level studies. Translational research is involving the techniques through which we can like save ourselves. Right. So it is like save ourselves from cancer. So it is like based on the solution. And clinical research is, as I already have told you, that it checks the efficacy and lethality of the medicine or the drug in the body that we are giving. And of course, lastly, you have the population research, which is mainly looking into the epidemiology. Okay, so it is like understanding where maximum cancer is happening, and how is this drug responding in that population. Okay, if it is failing, like why it is failing, how we can control. Okay, all these things are covered into the population research. And there are several institutes which are dealing with cancer research. So cancer research got a boost with the advent of CRISPR. Okay. Now this is the second booster that it is getting with the development of AI. So these are the list of the top cancer research labs in India. There are almost like 35 labs. These are just a mention of few. Okay. So on one side, CRISPR has revolutionized cancer research. Now it is a time when AI is going to revolutionize it. And there are several companies which are involved in cancer research in India. Like you have the Cipla, Dr. Reddy's, then you have the Zydus, Odon, Sun, Biocon, Glenmark, Lupin, Aurobindo, and Year India. And the key services they provide is like in developing the personalized medicine. Okay. They develop methods by which you can predict cancer before it occurs, so early detection, so that you can prevent cancer. Okay. They are also involved in drug development, mainly precise medications, and also involved in developing better diagnostic tools. Okay. And the salary range in these companies can start from 2 lakhs to 20 lakhs per annum, depending upon your skills, your work, your years of experience, and the position that you are holding. Okay. Now, as I said, AI is revolutionary, right? Revolutionizing everything. So where is this AI getting the information from? Well, from the electronic health records. So all that trials, the clinical trials, or all the research work that is going on, they are all getting recorded, right? So AI can draw information from there. The radiology provides a lot of information, pathology, molecular data, research data. Okay. And then they derive these data, they analyze these data, and they are capable of predicting the next clinical event, find similar patients, choose optimal treatment, and match to clinical trials. Now, here again, you see, like prediction of the clinical event, that how you can do? If you know how the cells are going to progress, okay, how the cancer cells are going to develop, what will be the next step, if that particular cell has the cancerous property or not, okay. So that early kind of detections can be done, okay. Now, suppose you want, you have developed a particular drug, okay. The trial, you have to select a group of patients or volunteers who are going to obviously test that drug, right? Now, this is a very critical step, because you have to find the correct category of patients, right? Suppose I have selected a drug, I have made a drug which is going to say have an impact in a pathway which is A, suppose, okay, A. Now you select patients, cancer patients, but over there, the cancer progression does not involve the pathway A, right? And you are giving the drug to be tested. Will you get some good results? Of course not, right? So here you need to obviously understand whether the patients that I'm selecting or the volunteers that I'm selecting, whether they have that pathway or not. Okay, so here comes AI. AI gets the record. Okay, it gets the record of the electronic records, radiological records, data, everything of those patients, and then it tells you that yes, this is the correct group of patients or not. Right? So that obviously increases your speed of analysis of trials. Choose optimal treatment and match to clinical trials. Okay, so one side is doing the research, another section is developing the drugs, another sector is checking whether we are doing it in the correct way or not, whether we can speed it up, what benefit we can get. Okay, and when all the three departments collaborate, you can really uh, you know, uh, resist cancer. So again, time to time, we are, seeing a growth in your these fields, the AI field, the clinical research fields. Okay. Now, clinical research, just let me give you a very small, short overview of this career, like what are the positions, how you can enter. Okay, so you first enter as a beginner. Okay, it can be as a clinical trial assistant or a clinical research coordinator, and then you get into the advanced course. So after, I mean, the second step, after 1 or 2 years of experience, you can become the clinical research associated, or you can study the startup specialist. You can become the regulatory affairs specialist. Then obviously you can become the senior and take up the management positions. So with further 2 to 5 years experience in these respective specialized roles, you can uh, you can become the team lead also. Okay. And there are various team lead projects which are available. Then obviously you with more and more experience and more skills, you get trans transited to director post. Okay, and you can also get into becoming an executive. Okay, so that all depends upon what is your skills, how efficiently you're doing your work, how you can improvise yourself. Now, to become a clinical researcher, you don't have to like do PhD or any such degrees. You don't have to get into that. If you're just a BSc or BTech or MSc MTech, okay, you can get trained yourself, okay, and join this domain of clinical research. You just have to come from a life science background. So it can be pharma background, can be biotech background, or any of the life science backgrounds, okay? The salary in this field is around, at entry level, you will get some around 3 to 6 lakhs per year. Senior positions will include like clinical research associates. You can earn around 5 to 12 lakhs, and director post, you can reach up to 25 lakhs or more annually. Okay. And the top institutes which are offering clinical research trainings will include your All India Institute of Medical Sciences, Indian Institute of Public Health, ICRI, Clini India, Indian Institute of Clinical Research and Management. Okay, there are several more. And the companies which are involved in clinical research in India, there are many. As you can see here, this is a just few list. There are many companies. Okay. And the key services they provide is like clinical trial design and management, regulatory consulting, patient recruitment and monitoring, data management and biostatistics. So you need to know some you need to have some knowledge in statistics as well, because you are dealing with data, right? you're dealing with the population. So you need to also equip yourself with statistical knowledge. Medical writing. Medical writing is very, very important. Many people think that, okay, medical writing is not that good as a job. But no, there is a huge demand for medical writing as well. Because whatever is coming up, that needs to be documented. Okay? So AI can also be used in this field as well, medical writing also. Bioavailability and bioequivalence and pharmacovigilance. Now, there are few high demanding roles. Okay, like the role of cancer genomic analyst. And what do they do? Analyze the NGS data to find cancer specific mutations and biomarkers. Okay, just a second. Data science can discover. Yes, Shraddha, definitely it can. Drug discovery, drug therapy, or therapy for cancer, these are all like, nowadays it is more like AI based. Nowadays what is happening more is people are first designing a drug, okay, based on the data available. They're designing the drug in silico. And then in the in silico manner only, they're testing it, whether it is applicable or not, whether it is good or not. And then they manufacture the drug, okay, and then they send it off for trial. So drug discovery, okay, that is also a very promising field nowadays. Yeah, previous slide, just a minute, Ashwarya. Here you go. This one did you want? You see, we may, as I told you at the very beginning, you can take up cancer genomic as a separate topic. You can take up clinical research as a separate topic, AI as a separate topic. In fact, few years back also, these subjects were considered as separate, but not now. Particularly after COVID, things have absolutely changed. I'll tell you one thing, like AI. AI played a very important role during COVID in developing the vaccine. Just can you imagine how could we get vaccine in such a short time? Where vaccines takes like around 5 to 6 years. Okay, because it has to undergo such tests and trials and everything. How did we get vaccines in such a short time in COVID? Thanks to AI. Thanks to some labs who developed those models by which you can test. So AI predicted what could be the, what could be the changes in COVID virus, okay, the predictable changes, because we saw that it was mutating constantly, right? So you are developing a drug today, tomorrow you're going to implement it. God knows, like it could have been that by the time you are implementing the drug, you are giving it to the population, the COVID virus has changed itself. It could have changed itself, right? It could have mutated itself. So in that case, the vaccine would have gone absolutely futile. You wouldn't have got any result, right? So those who were developing that vaccine had to keep that in mind as well. All right. So they used AI to predict all the possible mutations that the COVID can undergo, the virus can undergo, create a structure, design a model that could, cover all these different mutations. They used AI to select the population where this vaccine could be tested. Okay. Because as I told you, right, you have to, that selection of the population is very, very important to get the maximum result and get to the best results, right? So they used AI to select that population, and then they could uh, they got positive results, and then they released that vaccine in the market, and people got benefit. So this is about the vaccine development in COVID. Just imagine the AI power you can implement in any other field, like in cancer, in the neurodegenerative diseases, right? Could you share the top cancer research lab? Antara, can I just proceed? I'll come back to that slide. Okay? Just let me proceed. I'll surely come back to the top cancer research labs. That slide. Right. So basically, yeah, just one more thing I want to say, like, since I gave you the example of that vaccine. So what we can summarize from that particular example is that AI is helping us to speed up the trials. AI is helping us to make trials accessible. It is also helping us to like monitor the trials remotely. You don't have to go to the field all the time. You don't have to. And when I say field, it means like going to that place, that area. Okay. You don't have to do it. You can monitor it remotely also. And it is also helping us to develop personalized medicine. That is the power of AI. And it is developing. Okay. So those who don't take up AI right now will miss the train. It is gathering speed and it has a very promising future. Step onto the train right now or you're going to miss it. Okay. Coming to this role, what are the different roles which are there? So cancer genomic analyst, clinical data manager. So his role is to design, manage, and ensure quality of clinical trial data. Computational oncologist. Okay. So uses bioinformatics, ML AI to model cancer progression and treatment response. Molecular pathologist integrates molecular diagnostics into clinical pathology. And then you have AI research scientist, designs AI models to detect and predict cancer behavior. So to get into this field, what kind of educational qualification you need? Well, as I said, for clinical trials, right? Clinical research, bachelor degree is enough also. BSc, BTech, or you can go for MSc, MTech also. Obviously, see, if you have MSc, MTech, you will be, getting better opportunities, right? But BSc, BTech in biotechnology, molecular biology, bioinformatics, computer science. Okay. Masters in MSc, BTech, or even PG diploma in cancer biology, bioinformatics, genomics, data science, clinical research. Okay, those who are, even for nursing, those who are from the nursing background, they can also get into clinical research. All right. And of course, for those who are into the fundamental research, you have to go for the PhD, right? So cancer genomics, computational biology, translational medicine. These are the domains where you can do your PhD programs and, ultimately get into the companies and do some real work, real time analysis, right? What are the essential skill sets that you will be needing? Like some wet lab techniques you might be needing depending upon your role. Yes, Gunjan, you can definitely go into this field. So you just have to first understand which like which domain you want to get into. Okay, what role you want to get into. Right? So definitely you can look for it. And accordingly, you have to develop your skills. And yes, over here, let me tell you that Biotechnica does have a lot of these internship programs with hands-on and projects to develop your skills to help you progress in this field. Okay, there are clinical research trial internship. There is AI/ML internships. Okay, so you just can go to Biotechnica courses and you can check it out for yourself. Okay, to continue. The essential skill sets that you need, depending upon your your role, you might need to have the idea of DNA RNA extraction, which is absolutely basic. PCR, qPCR, CRISPR, flow cytometry, histopathology, NGS library preparation and sequencing. Okay, using Illumina and Nanopore. These are the latest ones. Bioinformatics and programming knowledge also, if you have, that is very good. So you should able to use certain tools which are very commonly used in this field. Like you have this GATK, okay, which is Genome Analysis Toolkit, IGV, RStudio, Bioconductor. You should be able to understand few languages, like learn new few languages like Python, R, okay, and also understand how to develop the pipelines. So you should know some RNA sequencing, WES, WGS analysis, variant calling, pathway analysis. So ma'am has already shown you some of it, right? She has already demonstrated these things. She has shown it all. And if you are like going into the clinical research, then obviously again, you should know some few have some knowledge in your GCP, ICH guidelines, FDA, EMA processes, informed consent, ethical consideration in human trials. Okay, the regulatory aspect. Those who are involved in the regulatory affairs, because all these field, okay, when you're working in this field, there is a section that is called the regulatory affairs. Okay. So you should know how to communicate with others, like other labs, other companies. Okay. And you should also look into the ethical aspects also. And few soft skills you should have, like scientific writing, data interpretation, interdisciplinary collaboration, okay, project management, communication with clinicals and researchers. So now, see, again, I'm telling you, these skill sets are is completely dependent upon like what you want to become, okay, which area you are choosing. And not only in India, like I showed you some companies in India who are working, and that the list is really quite big. But even globally, obviously, there's a huge opportunity to work on. Okay, US, Canada, Germany, UK, Singapore, they are all hiring uh, these genome uh, experts, the AI experts, the clinical research experts, the cancer research experts. Okay. And there are some global programs and fellowships as well. So in the end, what I can tell you is become an interdisciplinary expert. Okay. Like whichever background you are coming from, say, suppose you have come from BTech. Okay. Just you already know the basic concepts of biology, right? Why don't you learn AI? You can get into this field, right? Or you can just get yourself trained in clinical research, develop these skills, and then you can definitely get into clinical research as well. Cancer genomics is definitely one area where you can try out, because from the very fundamental aspect, what you already have, okay, from what you already know, the fundamental educational qualification, from there itself, you can get into the research of cancer genomics. Okay. But understanding the role of AI is very, very important. Okay. So start early with projects and internships, develop your skills, and stay updated on the trends, whatever is happening, like single cell, spatial omics, AI diagnostics, how it is working. Okay. Attend workshops, conferences, hackathons to develop your networks, right? And obviously, very importantly, stay among people who have the growth mindset, right? Because they can give you better informations and help you in your uh, career. So that's from my end. I'll just go back just a minute. I'm taking up your questions. Just let me go back to that slide you had asked for. Yeah, the cancer research labs. I have promised I'll come back here. Antara, here you go. So what do you feel after listening to all these opportunities? I hope I have been able to give you the overview complete. What do you think about this field? Yes, clinical research and all, they will hire you at the entry level also only. You can get in as a clinical research coordinator. Exactly, Abhay. This is a never-ending scope. It's opportunity driven. There's a huge, huge opportunity outside, and you don't have to like get some very high degrees to get into this field. You don't. You just have to master yourself in some skill sets. Okay, you're asking like computational oncologist, what skills you need to master?
Samrita ma'am, I'm so sorry to interrupt. Hello, Samrita ma'am.
Yes, ma'am, I can hear you.
Okay. So Samrita ma'am, I think you are done with your session, right? And our attendees are asking questions. So before we go into QA session, because guys, as I promised you all, we have kept a special QA session. Okay. And for that, all other speakers will also join us. And before that, we'll just take 10 minutes of time and we can have some quiz in between. Okay? Is it fine with you, Samrita ma'am?
Yes, ma'am, absolutely. That's a very good thing.
All right. Okay. So thank you
So
so much,
thank you.
Samrita ma'am.
Thank you, everyone.
Thank you, ma'am, for this amazing, insightful session, such informative informations that you have given to all our attendees. And I think all of them have made a note of it, and many people are already asking about the recording. So yes, you all will be getting the recording, okay, within a week or so for this entire webinar. Now, before we go ahead, yes, now is the time for quiz, okay? So I will display the QR code. Okay, so everybody please pay attention now. All the questions regarding the sessions, any queries you have with our speakers will have that after the quiz. Okay, now is the quiz time. And for that, actually, I'll be showing you the QR code. Scanning that through your phone, you can join the quiz. Okay. So I'll give you all 2 minutes to join the quiz and we can start. So you just have to scan it from your phone and attend the quiz from there. There are approximately, there are 20 questions. For every question, you get 20 seconds to answer. Okay. And the final result will be displayed on your screen on a leaderboard. Okay. Now, one catch is there that of first 100 registrants only can go for the quiz. Okay. So not registrants like for the webinar, but first 100 to enter the quiz only will get an opportunity to participate. So right now I can see there are 145 participants. So everybody, please be ready with your phone so that you come first and you get the opportunity to participate in the quiz. And our quiz will also come with a prize. Okay, what good is a competition if there is no prize with it? So the first person to get the highest score will be given a prize from Biotechnica. So at the end, when that leaderboard is displayed on your screen, so just take a screenshot of it and you have to forward it to cst@biotechnica.org to claim your first prize. Okay. So I will repeat once again, guys, that only first 100 people to enter the quiz will get the opportunity to participate in it. You just have to scan the QR code, and with that you can enter the quiz. Okay. At the end, you will get your position in the leaderboard and the winner. Okay. The winner will get a prize from Biotechnica. The first person to complete and to get the highest. Okay. Everybody clear with the instructions? Are you all ready? All right. So be ready with your phone, guys. I'll be sharing the QR code. Just 2 seconds. So here you go, guys. This is the QR code. Please scan it to enter the quiz. Okay, yes, I can see participants joining. Very good. Our slot is filling up fast. Yes, great. Yes, yes, I can see. I'm just giving 2 minutes for everybody to join so that we can start. Yes, few last spots are left, guys. Please join as soon as you can, as fast as you can. All right, so I think enough people have joined, and here you go. And here comes the quiz. Okay, time up. We'll have a look at the current leaderboard. Okay, good going. And here comes the next question. 20 seconds, guys, remember. Okay, so timed out happened. And the leaderboard has changed. So here we are going with the sixth question. Next question, here it comes. So Kasturi, you are there on the first place, doing good, keep doing it. And here we moving on to the next question. Okay, so guys, you have to be fast, okay? Yes. All right, next. Yeah, Gargi, you can catch up anytime. Sweetie James, if you have entered the quiz, then you have to go to the polls. Okay, you have entered the quiz, then you have to go to the polls on the top of your screen. Okay, Kasturi is still leading, Gargi and Sachi right behind her. Okay, let's move on. Next question. Still no change in the leaderboard. Okay, now we are halfway done through the quiz and we are at the 11th question. Great, guys. I'm happy to see so many people are actually giving the correct answer. So if we go for audience poll for any of these questions, audience is always winning. Very nice. Yes, come on, 4 more seconds for this question. Okay, some people got timed out here. Fine. Okay. So right now we have Kasturi at the first place, then Gargi and Sachi right behind her, followed by Sami and Tisha. Okay, guys, continue. That's okay. You can still participate, sweetie. Participate and feel that adrenaline rush. I hope all our participants are feeling that, right? Okay, don't have to answer here. All of you focus. Okay, next question coming up. Okay, so there is a change in the leaderboard now. Sami has come up. Sachi, you have gone little below. Okay, next question. Buckle up, everyone. Kasturi is still at the top. Srinidhi, I'm sorry if that is like you are getting this reloading message. You can try reloading, but I am not sure I can help you there. Okay, Kasturi, you have a competition now. Gargi has beaten you for the first spot. Okay, let's see who gets the winner's gift. Last 3 questions to go. Come on, come on. So you have to not only give the correct answer, you have to give it faster also. That is how the leaderboard decides. Okay, Kasturi, buckle up. Yeah, Gargi, come on. You cannot loosen up now. Last 2 questions. Suman, I think the registration, as I told you, that only 10 people, 100 people can enter the quiz. So that was done, and that's why you are not able to enter the quiz now. And anyways, we are at the second last question. So just 2 more minutes and we'll be done actually. So, okay, Kasturi has come up again on the first part. Okay, here you go, guys. Take a deep breath and here you get your last question. Awesome. Okay. So how was it, guys? I'll show you the leaderboard, but yes. How was it? Did you guys feel the adrenaline rush? Yes. Okay, okay, okay. All right. Let's see if there is any change. That was the challenge, Shraddha, because the questions were not that difficult. Okay, so the challenge was how to get the right answer, and that also in the least number of time. Yes. Yes. Okay. All right. Let's see who has got the first place. All right. So we have a winner here, and here it goes to Kasturi Sandhya with 5 minutes 23 seconds, and she has answered all 18 questions correctly. Gargi was very close, right behind her. But I think at the last question, Gargi, you got it wrong, isn't it? Otherwise, your scores were same. All right. Well tried, Kasturi. Well tried, everyone else also. Thank you so much for participating in such with so much enthusiasm. Thank you, everyone. Oh, okay. Don't worry, Gargi. You will get more chances like this with Biotechnica. We'll be coming up with more activities like that. Thank you so much, everyone, for your participation. And Kasturi, don't forget to take a screenshot of your leaderboard and send it to cst@biotechnica.org. Okay. All right. Fine. Okay. All right. So now all of you can take a breather. Okay. So our quiz is over. And now we are opening the floor for question answers. Okay. So all our faculties, all our speakers are here. So we have Divya ma'am, Nilofer ma'am, Somrita ma'am as well. And I think Kavita ma'am is not there. Okay. But she'll be joining shortly. Nurul Amin, the prize that's a surprise for the winner. Okay, so maybe next time when you win, you get a chance to have a look on that. Okay, something will be useful, something useful only for all you students. Like all our prizes are something which will be useful for you. Okay, all right. So we open the floor for question answers. Yes. Any kind of query, any questions you have, you are welcome to ask here. Okay, yes. So we have questions. Yeah. So Ritesh is asking a question in liquid biopsy, which precaution one has to take to do that? Divya ma'am, can you please help Ritesh in by answering this question?
Sorry, ma'am, could not get you.
The question is regarding liquid biopsy, like what kind of precautions one has to take for performing liquid biopsy.
Yeah, so for that, just a second. Yeah, for Ritesh, for that, the precautions or the best practices when you go for liquid biopsies, it includes few things like right from sample collection to timing of handling the components, then storage, centrifugation, everything has to be very careful. For example, when you go for sample collection, specifically for the cell-free DNA, ctDNA analysis and all, serum will be like least considered, and you can always go for plasma. Okay, because when you go for serum, you will have extra noises over there. So excess of DNA can be released and that can lead to some kind of clotting. Okay, so in that case, you can go for this plasma over the serum. Then whatever the sample you are taking, you have to process it as early as possible. Should not wait it for longer period of time, because when you keep on delaying now, there are chances of cell lysis and even the release of the contents from the non-tumor cells as well. Then whenever you go for the centrifugation part, try to separate the plasma. Okay. And the second will be to remove any kind of residual cells or any kind of debris. So in that case, what happens is that it will reduce the contamination part, and you can overcome that contamination part. Then apart from that, when you go for storage and allocating, I can say that please try to divide your content, your molecules, like your plasma content, into small aliquots so that it won't get contaminated. So whenever you require a particular ml, you can just take up one tube of that and you can go for it. Then apart from that, the regular lab techniques that we commonly follow, right? Whenever we are in the lab. So those regular routine works has to be considered. I hope so I can give you the answer, Ritesh.
Thank you, Divya ma'am. Yes, I'm very sure that he has got the answer because you are so elaborate in that.
Okay.
Okay, so next question we have for you, Nilofer ma'am. That is from Dr. Nagendra. Nilofer ma'am, are you there?
Yes, ma'am. I'm there.
Okay, so ma'am, there's a question from Dr. Nagendra, and he is asking that in a recent news, Russia came up with mRNA-based cancer vaccine named Enteromix. Is it going to bring the revolution in cancer vaccines? What do you think, ma'am?
Yeah, that is really true. Definitely, it will one of the very relevant method which is getting integrated, because if we all know that mRNA is one of the transcription process, okay, and there is a highest chances where we are looking for like how we can use this mRNA targets like with the molecules and then study the gene regulatory patterns, okay, irrespective towards the organisms, plants, and definitely a virus which is get you are targeting for. Okay, this definitely with the integration of AI will give a more predictive analysis, which we have discussed in the presentation also with one of the couple of examples. So basically AI with this being used with the miRNA or the microRNA targets ultimately help us to analyze the biological data sets and extract the meaningful insights, because here we are actually targeting that how the coding or else the non-coding RNA molecules are regulating the gene expressions level. Okay, and in that context we are able to understand many biological process which is happening, including the development and even with respect to the different oncological targets what we are looking for. And that exactly make us like to find the biomarker discovery, subtyping that's different oncological targets with miRNA biomarkers or as the liquid biospecie kind of stuff. And I think that is how we can look for the one of the potential identification is the identification of the prognostic mRNA targets. I hope that it give you an correct answer and even the overview for the same. Yeah.
Thank you, ma'am. Nilofer ma'am, thank you. Next, we have a question for Somrita ma'am. So, Somrita ma'am, there's a question from Azram. First of all, he's saying thank you for the workshop and he's interested in applying for a grant. So what would you say would be the most novel area or topic that truly addresses a gap in the field?
See, Azran, I can tell you one thing that over here, a lot of work is going on. So you literally have to go through the current research papers to understand the gap that is there. Okay, so read lots of papers and all the current papers mainly, and I'm sure you will understand what gap is there.
Thank you, ma'am. Yes. So Azram, yes. So see, when you are going to go in research, right, which is research. So the first step of that will be for you to do your own research to find out the area which interests you, which excites you, and where you think there is a gap and in which you can work. Okay. So your first step of research is doing that literature search, right? So you search through the literature as Samrita ma'am has suggested, and find out the areas, find out the things that you can, you feel that there is a gap, then you can talk to your guide regarding that, and accordingly you can proceed with your research.
Ma'am, I would like to add one more line to this,
Sure.
that
Sure.
when you are like going through the papers, you will also go through the techniques. So it will help you to understand the techniques by which the things are being carried out. So that will help you to design uh, the entire procedure of your work as well. So just like it's very important, like more than knowing, it's very important that you go through the papers first and foremost. Thank you.
Sorry, guys, I was on mute. Yeah, so thank you, ma'am. And so we are having such a huge number of questions. Yes. So next question we have for you again, Divya ma'am. So there's a question from Nurul Amin. So asking, adenoma is caused only in epithelial cells? Like, is adenoma caused only in epithelial cells?
See, what happens is that adenoma means what? It's basically a benign tumor. Okay, and it usually arises from epithelial tissues only. What you said is correct. So epithelial cells means what? They will be lining different organs, different glands, different surfaces in the body. So obviously it is not concerned with only one organ, but number of organs can be affected. Glands can be affected. For example, the very common sites are colon, rectum, pituitary gland, thyroid gland, even adrenal glands, breast, liver, all this can be affected. I hope so this answers your query, Nurul Amin.
Surely, I think it does. Thank you, ma'am. Yeah, so Nurul Amin has also responded. Yes, thank you, ma'am. So Divya ma'am, while you are here, so there's one more question for you. Like, I think many people are very much interested in knowing about liquid biopsy. So we have another question from Shruti Subhash, who is asking, can liquid biopsy give false positive result as well?
Yes, Shruti, there are high chances that false positive can happen in liquid biopsy because the main reason behind that is age-related mutation. So as we age, the mutation rates will increase. So basically, we call this as the clonal hematopoiesis. And it can happen also in non-cancer conditions like inflammations, even infections. They also release some kind of content into the bloodstream. DNA and other cell-free DNA and all. So definitely there can be technical errors arising out of that, which can obviously affect the sequencing as well as the sample handling process as well. But usually they are being confirmed with imaging and tissue biopsy before going for the treatment decisions. But still, yes, there are high chances of getting this false positive. I hope so I'm clear, Shruti.
Yes, ma'am. Thank you. Okay, so moving on, next question. Yes, so there's a career-related question also is there, and yeah, it's regarding the skills and all. So Nilofer ma'am, because like it's related to bio IT and all as well. So I would like to ask you, ma'am. This is a question from Sri Lakshmi. So MTech molecular medicine background, worked as clinical research coordinator for a few months, career gap of 10 plus years, currently upskilling in clinical genomics NGS through short workshops and courses, which is the right area to re-enter into career at the earliest and then develop genomic skills while working. Which particular skill should I focus for that? Nilofer ma'am, do you have some suggestions from Sri Lakshmi?
Yes, ma'am, sure. Okay. So, so as you have the experience with like a clinical kind of stuff, right? So my recommendation definitely will be like to explore some resources definitely with bioinformatics stuff because there is the exact data is get stored as I've mentioned in the presentation for the same. And then definitely, if you're also proceeding like looking for like how the clinical research can be get like implemented and how we can look for more predictive kind of analysis, because as I mentioned, like clinical is something where we are looking for actual uh, data, right? The real patient data we have, the every data point will have some correlations, and that makes to make a more predictive analysis, right? So it is a good to start with like clinical research domain. Okay. And side by side you can even explore the bioinformatics kind of what you can say as a course, beginner course of bioinformatics. So this two combination definitely give you an insights what exactly you can proceed for your further research or as a career plans accordingly. So then the bioinformatics itself have the different integrations, so that will come accordingly once you just proceed. I hope that makes sense, right?
Yeah, thank you, ma'am. So, ma'am, there is another related question. Okay, so this is especially for you also, because I know you are one of the key driving force of our Bio IT team and AIML course as well. So, this question from Sandhya. So, she is asking, as a biotech student, is it possible to learn AI and machine learning? I mean, will it be easy to learn? That's her question. So, Nilofer, ma'am, can you please answer
Sure.
her query?
Sure, ma'am, sure. So definitely it will be easy, because as I mentioned that in the practical, that if you are beginner also, right, in the programming stuff or ANML, you guys can definitely move on. Okay. Because while doing that kind of analysis practicals, right, you will, that kind of skills will automatically get like upgraded and build up, right? So definitely for biotechnological data analysis, this ANML and this bioinformatics, and definitely the coding is important. Because if you get, just remember, like, okay, so if you're working in the lab and you got to know like some novel findings, right, and you want to like showcase that finding, okay, correlate that findings by showcasing like with the integration of computational tools and the techniques. So why not to go with bioinformatics, right? Because bioinformatics is only what you can say as a inline process where you can showcase your significant results, and it's the best way to represent your findings. So definitely you can move ahead, okay, take help of the tools and techniques to analyze your big data analysis, right? And hand in hand integrate with ANML, so that the predictive analysis, okay, very targeted things you can get to know with the core research finding. So you will get both the expertise with your stuff. Okay. And if you have those both those expertise, so you are welcome in the industry then. Okay.
Yes, and Sandhya, one more thing is that in Biotechnica, we do run courses on AIML, and we have coding for biologists, all this certification internships we have, which comes along with the projects as well, okay, which will give you an opportunity to work in that project, get some hands-on experience. You will get to work with our instructors like Dr. Nilofer, and you will also get a chance to publish your papers as well. We give that kind of assistance also. So if any of you, if you are interested to explore this field of bio IT or bioinformatics, okay, so definitely you are welcome, and you can look at all the internships and the courses that Biotechnica offers here. Okay, moving on. Next question. Yes, so Samrita ma'am, there's a question for you. From Sweetie James. So she's asking, for people with employment gap and now shifting the domain completely to cancer genomics that aligns with the educational background, that is MSc in biotech, what are required bridging programs which will be required to get jobs in this field?
Yeah, actually, ma'am, this is one common question which I'm seeing in the chat. Many people are
Yeah.
asking about this, like they have a gap and they want to think. So I will say that yes, definitely you can come back to the to this domain. But since you have a gap, I would suggest that you do some kind of a course or a short hands-on training, and then to just uplift your resume and show that you are skilled, and then apply for the job. So in that case, you might be getting depending upon your skill, you can get into the basic level. They might take you in the basic level as a research coordinator or thing. And but then you can be very soon you can get posted to the higher levels as well. But since there is a gap, this is for everyone. my recommendation will be to do some kind of a internship, okay, with hands-on, particularly to be at a better position. And as ma'am has also mentioned that like in biotechno, we do have these courses. So you can look into these courses as well. The different, like with which domain you want to get into it, whether it is genomics, cancer genomics, whether it is AI based, you want to get skill yourself into the AI based, or whether it is clinical research, or whether it is regulatory affairs. Because all these, as I've mentioned, all these domains are interconnected. So you select your domain appropriately and do upskilling in that. That's it. Thank you.
Thank you, ma'am. Thank you. Thank you. I think your answer, it answers many students in the in our attendees here. Okay, so guys, we are running out of time. In fact, we have already overshoot our time, and we are down to last two questions. Okay, so the question that is there, yeah, Divya ma'am, it will be great if you could answer it. So this is from Gargi Panda. She is asking, so first of all, she has said thank you so much everyone for this amazing session. I'm curious, since tumor heterogeneity is such a major challenge in cancer genomics, how are researchers addressing it in sequencing data for personalized treatment? And in this context, how does single cell sequencing provide an advantage over bulk sequencing? Divya ma'am, can you throw some light in this?
Yes. So, Gargi, yes, what you said is correct. Actually, you know, in my session itself, I had given you a brief explanation that when we go for bulk sequencing, they analyze DNA or RNA from millions of cells together. So basically, we are getting an average signal that is going to hide cell-to-cell variability. Even the rare cell populations and even the mutations, early mutations are also masked. So in that case, we can go for the single cell sequencing because it's like a cell-to-cell resolution. So this will allow the researchers to map genetic mutation, transcriptional programs, even the drug responses of individual tumor cells. Not only that, it also helps to identify what I said in bulk sequencing, the rare mutations got missed out. But in this case, the rare mutations can also be identified. Like, for example, the drug-resistant clones, even the cancer stem cells that are usually masked in the regular sequencing method. So that can also be highlighted through the single sequencing method. So based on that, one can design this personalized treatment strategies so that they will not just the tumor cells they will be targeting, but they can also target all the dormant as well as the resistant population as well. Okay. Gargi.
Yeah, thank you so much, Divya ma'am, for this wonderful detailed explanation. Next question, Nilofer ma'am, we have from Nirulamin. Guys, I'm so sorry. I really apologize. I see so many questions coming up, and but this will be our last question. You know, all our technical team, all as well as our speakers, they are being online since 10 o'clock. Okay, all of you are all as well there. So we cannot extend the time anymore. We will. This will be our last question, and after that, we will be concluding the session. Whatever more questions you guys have, okay. So anything regarding yeah. So those questions you can forward it to our team, okay, and we will forward it to. You can send it to us, and we will forward it to our speakers, and they will be more than happy in replying you in email. Okay. So there's a last question, Nilofer ma'am, for you. Nurul she is asking in silico predictions of neoantigens for cancer immunotherapy are as reliable as experimental validation for cancer such as glioblastoma. If it is not, then what is lacking between in silico method and experimental validation? Nilofer ma'am.
Yes, ma'am, sure. So as the approach which has been mentioned over here, right, the neoantigens, right, which is very specific towards the analysis of the targets which we are looking for. So in silico study basically help you to identify first how the somatic mutations between the normal and the disease can be analyzed. Okay, so that is how you can target, and then you can look for like any kind of computational pipelines which help you to study the neoepitopes, okay, with the MHC binding affinity, and even look for the T cell receptors. So this is the approaches where you can target, and ultimately bioinformatics will help you with respect to like therapeutic developments and how to validate experimentally by studying the overall workflows and the patterns for the same. So I think that the things between like which you are looking for, that how the experimental validations is reliable, okay, with such kind of targets, and in silico will help you to bridge that gap, and then look towards like designing the pipeline and look for more accurate and predictive kind of stuff, and then move ahead for the validation. So I think this is how you can start with the processing step and bridge that gap.
Thank you, ma'am. Okay, now I will come forward and address one common question which is there in our audience, is how to get the recording and how to get the certificate. So everybody, yes, all of you who attended our workshop, you will be given a certificate for attending the workshop. Okay, and the certificate, it will be an e-certificate and it will be sent to you via your email. The recording also, as I told you, that it is this entire like 10 to 3:30, 3:45, the entire hours recording, right? It's a big, huge size. So we have to process it. Maybe we have to break it down. And then we will be sending it to you. Okay, so the recording of the workshop along with the certificate will be given to you within a week. All right. So with this, as we come to the close of today's national workshop on genomics, clinical research, and AI pathways to oncology careers, I would like to take a moment to reflect on the journey that we had together today. So as you remember, that we begin the day by understanding the basic fundamentals about cancer and how cancer happens and what are the different modern technologies like NGS, CRISPR, single cell sequencing, and how they are transforming the cancer research. Then we moved on to clinical impact, exploring how genetic testing, personalized medicine, genomics-driven trials are shaping the future of cancer care. Post-lunch, we witnessed how AI and machine learning are revolutionizing cancer research, from predicting progression and the drug response to integrating multi-omics data for precision insights. And finally, we explored career pathways in this evolving field, gaining clarity on the skills, opportunities, and global prospects available to aspiring professionals. Through animations, case studies, and hands-on demos, our speakers, they taught you, they told you, they gave you the information that was there in those particular sessions, right? We not only learned the science as a theory, we also saw its application in the real world. And then finally, we had our quiz. I hope all of you had a good time and participating in the quiz. And my apologies once again for those who could not participate. Maybe you have to be a little faster next time. And once again, congratulations to the winner, Sandhya. And the winner, so Sandhya, please don't forget to send the screenshot of leaderboard to cst@biotechnica.org to claim your prize. On behalf of the organizing team, I extend my heartfelt gratitude to all our distinguished speakers, Mrs. Divya, Ms. Kavita, Dr. Nilofer, and Mrs. Somrita, for sharing their expertise and inspiring us with their vision, and especially for joining us on a weekend. Thank you, ma'am. Thank you, everyone. So this workshop has been more than just a knowledge sharing platform, and it has been a step toward building a community of learners and innovators who will carry forward the mission of advancing career research and precision medicine. And last but never ever the least, a big thank you to all the enthusiastic participants for turning up on a weekend in such a huge number. You know, guys, at some point during the day, the total number of attendees for this particular webinar was around 225. Amazing, right? How 225 people from different parts of the globe, we came together and we shared our knowledge. A special thanks to all of you for your enthusiasm, curiosity, and active involvement throughout the session. Thank you, everyone, once again. And with that, I wish you all the very best in your academic and professional journeys. May today's insights guide you to ask better questions, seek deeper answers, and perhaps one day contribute to breakthroughs that change lives. Thank you, and we look forward to seeing you at our future programs. Thank you, everyone. Thank you, and have a great weekend ahead.